is a scale-up festival. We are located in Emeryville, California, and this is actually the building in which we are located. You can see our labs highlighted here on the third floor. Emeryville, California is home to many industrial biotech startups and mid-sized companies even, and we've been really fortunate to be a part of that ecosystem. This is a fisheye view picture of our fermentation suite. We start at 250 ml scale amber reactors and go up all the way to 300 liter scale. You can see pictures in the back there. We are supported by Department of Energy, EERE, through Bioenergy Technologies Office. We are primarily here to understand and solve scale-up challenges. So someone has a novel technology that was developed at milliliter scale, and they want to scale up to produce the first kilogram prototype. That's where we come in. We help them out through that process. Why do we exist? Those technologies that are developed at milliliter scale often do not perform in bioreactor with real-world feedstocks. The behavior is completely different. Culture experiences completely different conditions. So that's where we come in and we re-optimize the process. We also do quite a bit of downstream processing, and I'll go over that in a bit. We meet our collaborators where they are. Many of them start at the milliliter scale and come to us, but some of them have a decent sized process engineering team, in which case we would just directly jump to 300 liter scale with them. Most recently, we've invested in a pressurizable fermentation capacity, so to be able to simulate conditions in really large scale where the lower layers of the culture would experience substantial high pressure. And we've done a study with Checkerspot, which has been made public to show that their technology was able to, their culture was able to survive those high pressures. And we also do a bunch of chemistry upstream and downstream, start at the milliliter scale, go up to 200 liter scale with Hassel-Alloy reactors, which means we can use high and low acid concentrations. We can run vacuum, high pressures, a variety of different types of chemistry. We have a dozen or so downstream unit operations. Even then, we don't seem to have enough, in which case we rent equipment from our vendors. You see the italicized equipment here. They rent equipment to us, we install it, commission it, use the capability for our collaborators, and then unpack and send back to our vendors. We have core capabilities that we need at the ABPDU, but sometimes we do need these additional capabilities, in which case we would just rely on our parent organization, Lawrence Berkeley National Lab. If you're not familiar with the Lawrence Berkeley Lab and its user facilities, I would encourage you to look them up. You can engage with them directly as well, but we're happy to work with them to perhaps maybe understand a certain protein or learn its structure, etc. Molecular Foundry and Advanced Light Source are world-renowned for this type of work. If we were working together, today would be our first initial contact where you are meeting us. It usually takes about 12 weeks to start a project with us, especially only if you are a US company. If it's a foreign company or if a foreign national is involved in the equity of the company or at the C-suite level, perhaps in the management of the company, then it takes a little bit longer because we have to go through the foreign review process. But if you were a US company, this is our initial contact. We would walk you through, after doing the NDAs and reps and certs, we would do a project scoping. Project scoping would be at a very high resolution, about 4 hours per subtask. And that way we can be sure that the company is very aware of what are all the tasks that are being executed on the project. And then we go through the formal budgeting, contracting, and EHS review, substantial EHS review. It turns out that our EHS review actually helps companies long after they've left ABPDU as well. It helps them safely scale up their processes elsewhere too. This is an exceptional case in which we initiated a project within 6 weeks. We can't promise this every time, but if there is an urgency, like there was in the case of Hue, where Michelle Zhu wanted to demo some indigo dye coming out of a fermentation process in their demo day at Indy Bio Accelerator program, we were able to initiate the project, scale it up, and do downstream within 14 weeks. We were able to provide the indigo dye that went on an industrial machine. And you can see here Michelle is showing that to the audience. So this is how we helped 85 companies, more than 85 companies, small and large. We worked with the Totals of the world, the Shells of the world as well. So we're open to working with anyone. We worked with a bunch of academic institutes as well. So these are our academic collaborators. We support industry in writing support letters, co-authoring proposals to SBIR grants. Feel free to reach out if you're interested in that. We've helped put 17 products in the market. That's really our goal, is to accelerate the process of taking an innovation from a lab to putting products in the market. We conduct industry listening days. We've conducted 2 of these. These are not as frequent as the expos because we get a lot of feedback from our collaborators. It takes a little bit of time for us to catch up on it and provide the results, provide the feedback, use the contribution on the feedback. One example is gas fermentation capability. So the pressurizable reactors that I've mentioned, we've also been able to use these reactors with a manifold for many gases to conduct gas fermentation at half-liter scale. And now we're working with half a dozen or so companies that did tell us in 2019 to invest in this kind of capacity. We are now in the process of commissioning 20 liters of this gas fermentation capability. So we'll come to this topic today. ABPDU Virtual Expos, we conducted one last year, which was very successful. If you missed it, you can go to our ABPDU channel on YouTube. You go to YouTube, search ABPDU, and you'll find all the sessions at the Expo. Last year's theme was scale-up facilities, mostly scale-up and manufacturing facilities. This year's theme for us is software, equipment, and expertise. So if you missed yesterday, we had a whole day of software, data, and AI talks, some wonderful talks, and we recorded these. Unfortunately, we cannot put them online immediately. They have to go through a review process, so it'll be a couple of months if you've missed it, but you can find them on our YouTube channel in the new year. Today, we are going to start off with equipment, mostly from our collaborators from Europe, because we want to give them a chance to present in a decent hour. Then we go into expertise, again start with our European collaborators and move into the American contributors, and then eventually we'll go into equipment. So quick instructions. Please stay muted at all time unless asked by a moderator, whether you're a speaker or not. Please stay muted until you're asked to. This is not a webinar format. This is a direct meeting format, so we request you to stay muted. If you have any technical issues, you have any questions or concerns that you need to share, please write to us at abpdu@lbl.gov. 6 of us are monitoring this email ID so we can respond to you immediately. If you have any questions, please write those, or comments, please write those in the chat section. The speakers are expecting that you will write the questions in throughout the talk, so you don't have to wait until the end of the talk. So I'll stop there and very quickly thank the ABPDU team that does the fantastic work of the scale-up and develop the scale-up expertise that we have. And very quickly, any questions, comments, or concerns? All over to you, Nick.
Thanks, Deepti. Welcome to this session, everyone. So our first speaker is Christine Munch from Enduro Genetics. The topic is about bioproduction at scale. Christine, over to you.
Thank you very much, Ning, and thank you for the introduction, and thank you for the invitation, Deepti, and so forth. Give me just a second to get up my presentation. Thank you very much. Yeah, as I said, thank you for having us. My name is Christian Munk, and I'm with Enduro Genetics. And at Enduro Genetics, we are all about bioproduction at scale. What I would like to cover today is talk a little bit about the sort of the opportunity that we see in what we address. You could say this is also the problem that we solve. The science behind the solution that we have, what it is that we actually do in terms of implementation and how we use our technology and the impact of that, and then value creation when we look at both from a customer and from a bioprocess standpoint. Maybe as a brief introduction, also we're based in Denmark, and we are just short of 20 people here, and we work with a number of clients across biomanufacturing and bioprocessing at different scales, and also at so both early startups, but also large companies, and across the pharma companies, and also what we call biosolutions or bioindustrials. So maybe I'll just use that as a prelude. But if we look at the core of what we're focusing on, it's really about solving the problem about cellular or cell-to-cell variation or cellular heterogeneity. As a lot of us know, when we work with biofermentation, then large-scale fermentation, it puts a burden on the producing cells, and that results in cellular heterogeneity. If you look at the drawings to the left, it's a very good representation of what goes on. You start out from the laboratory, and you've engineered a cell. You've done all your metabolic pathway engineering work, and you have the ideal clone, the ideal single colony that you then start to scale up. And what happens as you scale and as you move from a single colony all the way up to large-scale fermentation is that using a production host, bacteria or microorganisms or a yeast or bacteria, it's a burden, and the microorganisms will start to evolve, and new mutants and non-producing variants are being produced. If you look at the depiction to the right, it's a more simple illustration, but what it shows is that over the many cell generations that happens, eventually the producing cells and the cell population will shift towards a non-producing cell population. Excuse me. How fast that happens and at what scale depends on the burden, and also on the production system, the production host organisms that you're using. But we see again and again across the cases and the clients we work with that it's an industry-wide problem. If we look at the solution that we work with and how it works, then we work with a technology that we call EnduroSense. It's a genetic biosensor plugin, and we use it to couple cell health to production, high production. And as we say, is we can make the cells addict to being in a high productive state. On the left of my slide, you see a depiction of a very simple depiction of a cell that produces a product. This would be your production host organism. And the way our technology works is that it's able to sense when a cell is in a productive state, because that has a transcriptional state. This technology acts as a biosensor, and it's coupled up next to an essential gene in the production host that then regulates the growth. And effectively, what it means is that only cells that are producing or high producing are able to efficiently grow. If a cell deviates from being in a productive state, as a mutant cell is formed, then its growth is slowed down and is eventually being washed away. So what it means is that you, at any time in the process, as you go from your plate all the way up to production, will always have a cell population comprising of high producers. It's a technology that's agnostic in terms of products and also production hosts. Today, we work with a number of different production systems: E. coli, Bacillus subtilis, different type of yeast, both piquia and baker's yeast. And we've also worked with the filamentous fungi like Aspergillus oryzae. The technology is it's a plug-in technology, so you can plug it into your existing production host, and it works irrespective of your media, and there's no additions that you need to make. It's not being induced. So it's a one-time plugin, and it's a plugin that you do to your production host. So that also means that it can be both applied in processes, in production hosts where you're scaling up, as well as existing production processes that you have on the market. the regulatory burden in terms of implementing it is very, very, very light. The impact of then implementing it looks like this. This is the type of plots that we typically see. In the yellow line, you see a conventional production declining curve where productivity of the cell culture sort of drops over the different generations and as you scale. In the blue, you see an example of a plot that we typically generate. And typically we see two effects. We see an A effect, which is a baseline tidal lift. This is simply because, or not simply, but it is because right the way from you take the colony sort of off the plate up until you're in production, you prevent the emergence of low producing or non-producing cell variants. And then as you scale over generations, you also prevent the decline and the further fermentation of low producing cell variants. If you look at value creation and also where our technology is typically being applied, then this to the left you see a sort of a graphical depiction of a typical S-curve of how you move from research to scale up and eventually to production in a biomanufacturing process. We work a lot with clients who are in production and who wants to take an already optimized process and optimize it even further. And they might even have a good understanding of the cell heterogeneity and the amount of non-producers or low producers they have in their cell culture. This is a typical use example that we do, and there we can deliver tighter improvements and help our clients become more productive in terms of that. You can also use it in your scale-up journey when you actually have something in the lab that might work at lab scale bioprocessing or bioreactor level. But if you start to scale up beyond that, you might have problems remaining productivity. This is, so crossing this valley of death that a lot of companies in. We also have cases of that where you apply the technology and then you can scale it up. There's also emerging applications where we use it for A-like applications. This is a little bit more early stage, but we use the technology to sort of self-select towards higher productivity over time, doing that optimization in the bioreactor. So the value creation from using our technology is a lot around increasing the final titers for products that you have in. You can also use it to strengthen the tolerance levels. So we've been seeing some use cases for that also. It might be cases where the product itself is toxic to the cell. So here you can increase the toxic tolerance by selecting for phenotypes that are toxic to that molecule without them dropping productivity. You can have a more stable process over time, and that opens up for certainly doing extended fed batch fermentations, or these fill and draw fermentations, or even continuous fermentations. We have clients that are looking into that, and of course you have a baseline tidal lift as which is always worth something when you talk about productivity, because right from the get-go you have a more high producing cell culture. If we look at the area where we focus a lot, as I said early on, it is really on helping already established, optimized processes become even more productive. And a depiction of the type of value that we create here is what you see to the left, because our technology works by as it works by changing the cell population. You don't change the inputs. So you could say simply put, with our technology and a bigger population of high producing cells in your cell culture, you have the same amount of input, but you have less energy carbon going to producing biomass of non-producing cells and more towards producing product. which of course can mean a lot. Typically, we see titer increases of 20, 25, 30 or above increases for already optimized processes, and more if it's more emerging processes. And if you start to play around with production time and other things, there's just ways to optimize the process. And on concluding remarks, being respectful of my time starter here also, I think the key message from us is that biological production is a burden on these cells. So they want to grow, they don't want to produce, and nature finds a way to grow and not produce, and that's why we all have the problem with the cell-to-cell variation. And EnduroSense is a way to increase productivity at scale by addicting these cells to high production and leveraging the natural evolution that you already have, just directing it towards high production. It works in any organism and at any scale, and it also works for any product. And with that, I will conclude my presentation and take a question or two. Yeah.
Thank you, Christian. Actually, you have half an hour instead of 15 minutes. So we have a lot of time for discussion and questions. Just a reminder for everyone, if you have questions for the speaker, please put it in the chat. So I have a question, Christian. Can you talk about the specific scale Enduro can work with?
Yeah, I can. So at our own facilities here in Copenhagen, we can take in client strains and then maybe I can just describe our workflow and then I'll talk about scale. So when we address a client project, then typically what we do is that we start out with samples from the fermentation where we do RNA-seq analysis on. And we use that to define and understand what transcriptional state the process is and the cell population is over time, and we correlate that with the production curve. And that allows us to understand what state is the cell is a high producing cell and what is a low producing cell. So we define that, we use that to define what good looks like. Then we use that information. So that of course requires that you have a bioprocess already, and it only needs to be a 1 liter or small scale bioprocess. It does not need to be large scale, but it can also be large scale, and we also do it up for that, of course. Then we use that data to design computationally and with our algorithms EnduroSense constructs. These are the constructs where we couple different type of sensors with different type of essential genes to create some diversity around that, and then create a library of strains that are then being screened. And they're being screened in a very classical way, using typically our clients' protocols for that in terms of how they measure productivity on the product of interest. Then at our own facilities here, we can also do up to 1 liter bioprocessing validation. And if we go beyond that, we have partners we work with, including you, that we work with to demonstrate it. And this here, it's everything from 50 liter up to, I think we did 300 liters with you that we've done. Very often clients want to do that themselves. They as soon as it sort of leaves the bench, the clients often almost always have their own production pilot facility or their own protocols for how they want to test that. So we typically don't do that as much.
Got it. Thank you. Do you mind give us some examples of a specific process you have worked with?
I can give you maybe some examples of I can give you some of our own case examples. So an interesting case example that we worked with was BLG, so beta-lactoglobulin. We worked a lot with that. So that's a protein of interest that's very interesting for it's cow milk protein. So we did our own case example because it was so we saw a lot of interest for it, so to say. And there we did a couple of things. We both demonstrated that you could use it to sort of solve the scale-up gap, which is actually also part of what we showed with you, where you had a process that was stable and productive at 1 liter, but as soon as you moved it to 300 liter, the ancestral host, the control, as I called here, was not stable. It didn't scale. With our plugin technology, it suddenly scaled. So that's a sort of a case example of where you can quickly move to that. We were also able to show that you could do continuous sort of a chemostat continuous production over several weeks, several weeks, where the control strains dropped after 72-ish hours, and we managed to run it for several weeks, and eventually we actually needed the equipment for something else, so we had to take it down. But I think that's one case example that we've used it for. We've done a lot of case examples also in E. coli on different type of both working with clients and also working on our own case examples. Some of the case examples we've done there have also been on compounds that are sort of semi-toxic to the host to do that. We worked with a Cerevisia, so baker. She's also demonstrating a pharma a peptide used for pharma applications, where we did our own demonstrations there to show that we could do a continuous type fermentation. And then we have a lot of client work that we can't sort of dive into, of course.
Great. Any thoughts for the downstream processing, like when you do the scale up, right, of about bioprocessing, including both fermentation and downstream? What's your thoughts? Like, how do you think about the challenge for scaling up the downstream processing?
Yeah, I think as we work with clients, we're learning more about what's important in downstream processing. So, for example, the less solids you have, the better is what we're learning, of course. And I mean that's intuitive, but we're not downstream processing experts from our side. So we're seeing that as something we do. And then I think also, I mean, the more I think we're learning as we work with clients. That the more we know about the entire process, the better we can actually tailor the solution to what we do. Because really what our technology can do is within a population, if you can define a specific phenotype that you like, this is what good looks like, then you can use our technology to drive the population towards that phenotype and prevent genetic escape at the same time. And and of course, high productivity is a good phenotype, but there could also be other phenotypic traits that you might have. Could be, certain type of tolerances to either to the environment of the product of interest and these things, where again, from transcriptional state, it can do that. Yeah.
All right, thank you. Any other questions from the audience? Thank you again, Christine.
Thank you all. Thank you.
Do we have Kamila?
Hello.
Hello.
Good morning.
I was wondering.
Good morning.
Yeah. Morning.
Can you hear me well?
Yes.
It's okay
yes.
with the audio? Perfect.
Great. So Kamila Beha from TetraPak. The topic will be on innovation for new food sources. Kamila, over to you.
So good morning, everyone. So my name is Kamila Abelha. I'm responsible for the sales and business development department in the business unit that we call New Food at TetraPak. And I'm going to give a brief introduction of our company, and then gradually I move to the topic that is how we are acting or how we are playing or supporting projects and customers that are in the new food fermentation area. So we are a company, family-owned company, and this is a bit about our figures. We're still with the figures from 2023, so we are updating the figures of 2024, but I would say we are present in, to highlight some of the points here, we are a company especially known by our aseptic packaging solutions. So you see here that we have delivered in 2023 193 billion packages globally. And also we are almost 25,000 employees globally in the company, and we are present in more than 160 countries. So we also have, besides the packaging business, we have the processing part, where we have technologies that are suitable for both the food products production, but also for what we call ingredients related to processing under precision or biomass fermentation. So this is a bit about what we are going to talk further. So besides the processing and the packaging, we have also the services part, which is quite important, let's say, especially when we are implementing a solution, a line solution, a bioprocess line at our customers, that later on we continue to support them with our services. So we have a wide global after-sales presence in several of these 160 countries. We have the coverage of those countries by our after-sales and services department. This is a bit more into what we are in the processing. So numbers here that are perhaps interesting to highlight is that in terms of unit operations, we have more than 104,000 units delivered globally. And in 2023, we have made 1.5 billion euros globally on sales of processing equipment related to the technologies we have in our portfolio. And we have been present, in the markets for almost 70 years. So this year we are completing 70 years of presence as a company in the processing solutions area. Our, let's say, way of expressing what is the company about is to talk. We are one of the world leaders on food processing and packaging solutions. And also, again, we always look into how we can closely support our customers, as well collaborate with our suppliers, meaning aiming to have safe, innovative, and environmentally sound products that can support people or feed people across the globe on those, again, these 160 countries that we are present. Our vision or our purpose is to make food safe and available everywhere, and still doing this by protecting the planet and make food people having access everywhere to products for consumption. Looking to the future, so a bit to explain how has been our journey to start to look into other processes besides the food processing part, looking to more this, again, process solutions or bioprocess solutions for biomass or precision fermentation. If we look a bit about the situation, how is forecasted future for the planet, we need more food by 2050, So we need approximately 60% more food available for the growing population. And we are going to be almost 10 billion people on the planet. And the land, the limitation we have is how much land is available to produce food, especially to generate proteins for human consumption. And another point is how we protect the planet. So we need to be mindful on how to protect the planet when it comes to greenhouse gas emissions. So the combination of these factors brought to us how we can think about solutions to continue to provide food safe everywhere. What can be from our side done to secure the future for the population that is growing. You see here we have basically 4 pillars that are towards to our strategy to support the growing population. So we need to find ways to enable a more sustainable dairy. So how we can continue to support our existing customers that are mainly in the dairy business to reduce the greenhouse gas emissions from their production of food, as well ourselves, how we can be mindful when we are producing our equipment for food processing, how can we be mindful also to protect the planet? So how we can work to reduce the greenhouse gas emissions from our processing equipment. So this is one pillar that we are taking actions. Also, the second pillar, that is the one that's more related to what we are talking today, is how to find new solutions for processing food or to enable the process of food. Finally, here, I think it's not so much, and now it's better. So you also need to find the solutions to reduce the food loss and waste. We know, for instance, in US, we have almost 38% of the food that's produced is wasted. So we need to be mindful also on how can we contribute to reduce the production loss. So we have a commitment, and this is the targets we have by 2030, that is to contribute from our side to reduce the product loss in 50%. And also, not less important, but it's also about how to have more food available, how to scale the access to safe nutrition through sustainable food and packaging. And this is about how we can make organic grow that still we can provide or make food safe everywhere. The pathways, I mean, they are all somehow linked. So those pillars that we have been talking, they are very much linked to each other. So, and then when we convert then into what we can provide, we also have a very important pillar, that is the automation and digitalization. So how can we make solutions that are intelligent solutions for manufacturing food products and lowering TCO to our customers? So how to combine automation and digitalization to the processing or bioprocessing solutions we are meant to support and provide to our customers. Talking about fermentation as such, this is to illustrate some points that we need to be mindful about. So we have to the left-hand side one very important factor, that is how to make a scale up, how to bring facilities that are from lab to pilot and then to industrial scale. Also, how we can make intelligent, cost-effective design for the fermentation, for the bioreactors, automation, and how we can combine the academia, the contribution of academia, and also the recipe development. And then at the center part, we have a bit on the techniques that we are talking here, like especially the precision fermentation for the production of certain specific ingredients. The technique as such is not new. It's something that it's already from long time being used in the pharma industry, as well to the production of certain ingredients that are today utilized on food production. As an example, rennet, that's an ingredient for the cheese production. This is also coming from fermentation techniques that now are moving towards to be more utilized on producing ingredients that are meant to be part of food products. And then there is this relation that we see from the pharma perspective. When we were producing ingredients, we have low volumes with high value, added value products, end products for the pharmaceutical environment or for the space. And then when it comes to food ingredients, then it's a bit how can we make more cost effective? So we need to deliver more volume to be able to, back to the growing population, to deliver the amount of food that the growing population is going to require by end of by 2050, right? So we need to think about volumes that are much bigger than what the techniques were delivering under the pharma industry, but we also need to be mindful on the high added value that is not there necessarily on the ingredients. So we need to make these ingredients cost effective for them to make their role on still providing food enough for the planet in a sustainable way. And then also we talk here, when it comes to food, we have the angle of ingredients, but we have also the industrial biotech. As TetraPak, we are very much recognized from especially from the processing part on our expertise, having the technologies and how to provide solutions, complete line solutions for processing food. So we are very much known by the food categories that are represented here from left to right. So 6 food categories. And then we have the new food category that's specifically dedicated for bioprocess solutions under biomass and precision fermentation applications. And of course, the combination of the knowledge that we have in food categories in regards to application and end functionalities that we foresee in products and how to process end products, this is also helping. It's a synergy to what we need to do when it comes to produce new products based on ingredients that are coming from new food, so from the precision or biomass fermentation lines. So we emphasize that we can combine the expertise of being able to provide bioprocessing line solutions, but also assistance to all the way down to the end product. So how to formulate, how to develop, and how to process products with ingredients that are coming from new food. From timeline perspective, of course, where we place where we are today is, of course, focusing on the fermentation derived protein sources. So we had, backwards, we have the animal-based proteins as a main source for human consumption. Then we had the plant-based that is still growing. There is still some developments ongoing, including in our, let's say, in our area of expertise in the company. We have a lot of projects in regards to plant-based proteins, on how to process, how to convert the sources into end products for end consumers. And then the next, where we are today, is with the fermentation derivative. And we know that there is other trends related to cultivated meat and insect-based. So this is also evolving, and mostly likely we are going to be in the next focus, or the focus of our company is going to go on those technologies as well. Technology-wise, we have inside our portfolio of processing, we have more than 200 unit operations, which the technology behind is developed by ourselves. And under those technologies, of course, what we leverage when it comes to bioprocess solutions is how we can utilize or we can capitalize on those technologies to build up bioprocess line solutions. So how we need to understand what is the required functionalities, the level of purity that we need to from fermentation all the way down to the end of the downstream process, how we can leverage our knowledge to bring productivity, to make the production of the ingredients still to the hygienic level that we have. We secure that the equipment, the unit operations that are part of the bioprocess line, they are cleanable, that we can perform CIP and achieve the right level of cleanliness. So we leverage more or less on the knowledge that we have of almost 70 years providing solutions for the food industry to utilize also this knowledge, utilizing our portfolio to creating line solutions in bioprocess. Here you see a bit of, it's a kind of overview of the different unit operations, the different technologies that we have under our portfolio. And then the other factor that is important is we have the knowledge on how to understand what are the crucial parameters from any bioprocess that we need to capture, that we need to base the solution, the scale up to provide a solution at industrial scale. So one of the key areas of expertise for us is also that we know very much our technologies and how from a pilot scale to industrial scale, which type of process we need or bioprocess data we need to access and which type of data we need still to work to be able to design a line at industrial scale. So this is both upstream, downstream, and also on the fermentation process. Fermentation process, of course, is the core of the line, but then again, the productivity that is essential to come out from the fermentation needs to be also supported by what is happening upstream and downstream. And as I said before, there are some key parameters that we have quite a lot of knowledge and experience that, especially, let's say, upstream, we have been one of the leaders on the industry on knowing aseptic technology, sterilization processes, so how to design sterilization units for heat treatment of food products, but this is also where we leverage to be able to do heat treatment of growth media, and how to make the link for the sterilization process towards to what needs to happen in the fermentation in a safe way. So having the aseptic design in place. Moving to downstream, mindful on food safety, cleanability, because we, like I said before, on the downstream process, we need to make a solution that fits for the purpose. So how much purification, as an example, we need to have of an ingredient for the ingredient to perform as expected in an end product formulation. So where we leverage the fit for purpose solution that still is cost effective and is delivering what is expected at the end of the line, moving forward to our end product. This is an example on the building blocks that we might have part in a precision fermentation line. So for the building blocks, we can see on the bottom of this slide what are the technologies, roughly. Some is a kind of illustration, some few examples, but for more or less every step that we have upstream or downstream, we have technology portfolio to support. So we can be the ones providing the line solution. And moving forward, let's say we come to an ingredient as we see to the right-hand side at the end of the process. And this ingredient, we will be able also, if it's B2B or B2C, we will be able also to understand and support with our technology knowledge, application knowledge on how to formulate products utilizing that ingredient. And backwards also give indications if certain functionalities that were expected to be delivered by that ingredient in a certain application, if they are not happening, we can always give indications to what eventually needs to be reviewed in the downstream process or in the fermentation that can bring back what is expected to be in the ingredient. Another point that is I would say it's value from our side that has been recognized by our customers is our capabilities and our know-how on implementing what we call large or complex projects. So with the organization that we have for project implementation and by the methodology that is applied for project implementation, we have the excellence to also not only design the solution, but also moving forward, make the detail engineering, make the design for installation, do the installation, do the commissioning, and they all way to put the line in commercial production. So we can work from different perspectives to deliver the full solution. So it's what we state as a solution provider. So it's not only about designing, but it's also knowing how to make it work in an efficient way and be there, especially moving forward to support, to secure that our customers are producing as they were expecting when they came initially with idea what they were meant to produce, all the way to what needs to be, let's say, performance at the end. And also be a bit elaborating a bit what we see here in the wheel. So the methodology, of course, I'm not going into the details of the project execution methodology, but we work very close to our customers and also with the partners. So eventually the full integration of the bioprocess line along with utilities, along with civil, with the building, etc. We have the knowledge to interact and to make those puzzles to come together to make the installation performance to come as discussed, as agreed with our customers. And also I think how to implement the project, how to understand what are the critical paths, what needs to be the key milestones for us to take decisions along with the project implementation. This is also the way we foresee always to support our customers with our experience, with our view on what needs to be decided today for the production to be running from 1st of January 2026. So we can identify already today everything that needs to be decided tomorrow. So we can support our customers on that. And not least, not so less important, but also to highlight here is how we support our customers on validating how the unit operations, the different unit operations they should perform as per design, as per the inputs that we have received to design the units, and that they all combined, they are synchronized to deliver the productivity, the yield that is expected at the end of the bioprocess line. Three different pillars three different approaches here on how we can work together on, and still our main mandate here is quality assurance. So we can, of course, support fully with the design. So if it's a project that there is not so much experience on how the design should look like, we have the engineering capabilities to support from the idea into a concrete proposal, aligned solution. We can also work together because we also leverage and we also respect a lot the knowledge that the players in the field, they have about the process. So we also join forces that we combine the knowledge from both sides to make the design of the bioprocess line. Or we can have the third pillar that it's very much the case, or can be the case, that is our customers, they have much more understanding and experience with the requirements, with the necessary inputs to design the line. So they are specifying and we are executing and securing quality according to their expectations. Just a bit about an overview on how a project organization looks like. And I think the point here that's important to highlight is that we have a governance on how we run projects. So we have regular board meetings to support and validate the different toll gates that are under this project execution methodology. And this, back again, is to secure quality of what we are meant to deliver at the end of the timeline of the project. So we have certain milestones that are connected to our internal governance that we need to follow. And as well, those toll gates, they are linked to the validations that we jointly do with the customers to make sure that every milestone up to that specific toll gate, they have been fulfilled according to the customer expectations. So the transparency is, I would say, what we have as a mandate when it comes to how we are executing the project and how we are performing every single milestone that needs to be in place for the project to be concluded. The coverage, so we have what we call the project centers, where we have our project implementation capabilities, excellence. So we have across the globe 16 project centers, as you can see here in the map. So this is very much also related on how we can cover globally with our knowledge, our experience on implementing projects, like I said before, complex, that's not necessarily a large project, but large projects that they can be even around $200, $250 million. So we have been implementing projects in the past few years around this size. Last part, I will try to be quick for questions. So it's about, we cannot do ourselves alone. So we are also leveraging in partnerships, in collaborations across academia. And also we are constantly looking to how we can leverage or build more knowledge on the applications that we are working. So it's not different from what we have been doing in the food applications, but is also valid for the applications under, you're into precision in biomass. So we have in Sweden an initiative that's called Biotech Heights, and this is in partnership with the Lund University. And this is very much that we start to support players jointly, that they have an idea, they are at a very early stage of what they see that needs to be their process. So we start at very early scale, lab scale, as you can see on the bottom part, left side, half liter is the smallest bioreactor that we have available in those facilities. And what is important to highlight here, you see on the right side, all the partners that are collaborating with us in this ecosystem. So it's of course driven by TetraPak and Lund University jointly, supported by the government, the Swedish government, but we have plenty of other partners that are supporting ideas to come up to a pilot scale. So this is something we are very proud that we are implementing with many of the players, that they have great ideas, innovative ideas, to make them to become a reality. We also have a small facility inside the Lund University. That's not exactly what I show in this slide, but it's a research lab that is also for us to close to the academia, the collaboration with academia, understand what we need to learn next. So it's more looking towards to the trends and what we need to look for the future, for us to be able to support with solutions that are at the right time to customers. And we have finally our pilot facility that's going to be inaugurated. It's also in Sweden. It's going to be inaugurated now in 2025. And we have capabilities to run full bioprocess fermentation and precision fermentation processes. So the main purpose is also to help our customers that are in a pilot scale to come to the scale up into industrial scale. So this is my last word is about what we do in a create an ingredient in this pilot facility that is going to be inaugurated in 2025, we can also leverage on our product development centers to understand in the end application how this ingredient is performing. So we can combine both capabilities with our product development centers to understand, as I said before, how the ingredient is performing and what we can do to leverage or to improve the performance if it's not yet what our customers were expecting. So then I think I close here, and then I can open for questions. Widening. Sorry.
No problem. Thank you, Kamila, for the comprehensive overview of your company. It's very impressive.
You're welcome.
So because we are on time for the next presentation, let's move the Q&A to the chat. If you have any question for Kamila, please put your question in the chat. Kamila, if you can stick around and answer questions, that would be great.
Sure, thank you. Then I do on the chat. And thank you very much for your time, everyone, for participating. And I wish you a nice day and nice conference. Thank you.
All right, let's welcome our next speaker, Antoine Charbonneau from Applexion. And the topic is about scale-up equipment and expertise. All right, Antoine,
hey everybody.
take it over.
Can you hear me okay?
Yeah, I can hear you.
Can you see my screen?
Yes.
Okay, perfect. So good morning, everybody. My name is Antoine Charbonneau. I work for Applexion. So it's formerly a company that was part of the group Novacep, for the ones who know. And Applexion is a French company. I'm personally based out of Denver in Colorado in the US. So maybe I start with the vision of the company. It is really to contribute to reduce the climate impact for our customers. We increase the efficiency of their daily production. You'll see later. I will go a bit more into details about that. And we really try to develop processes for healthy food and for the new biosourced molecules. Our purpose is really to offer day-to-day solutions for excellence in purification processes. That's really what we do. We are the core of DSP, purifying liquids on day-to-day. So about the company itself, so it's headquartered in France, in Lyon. But we also have another technical and sales large office in China, in Shanghai, where I worked for a few years in the past. And also another one in partnership with the IBRL that I'm sure a lot of you guys know here, of the University of Illinois. And we have also other offices around the world. Worldwide, we have 150 people working for the company. 10% of them are dedicated to the R&D in the different technical offices we have all around the world. We typically install 30 to 35 industrial equipment worldwide every year, and 5 to 10 pilot units, and we also revamp existing units so that they work better and they have a less of an impact on the environment. So it's always a chicken and egg because we about the market and the technologies. But today I will take the chicken and egg, starting with the markets. Well, the company was founded in 1968, so almost 60 years now, in France. And the name Applexion comes from, actually, it means application in ion exchange. That's where it comes from. Please don't say Apple X ion, because a certain phone manufacturers doesn't like that their name is within our own name. So it's Applexion. And at the time, in 1968, it was really about addressing the market of the food markets. The sugar, in France, we do sugar with sugar beet mainly. And you need the juice, the beet juice, to be decalcified, softened, in order to protect the evaporators. But also the dairy industry that is pretty big in France historically. You know how much French people love their cheese, right? So that's how it started. Ion exchange in cheese and sugar. And then as the food market evolved, I would say, new opportunities arrived, and also the company increased the amount of technologies in its own portfolio. So we started dealing with what we call functional ingredients, like fructooligosaccharides, galactooligosaccharides, inulin, fibers, plant proteins, also all the new sugars that are used in order to help tackling the health issues we have linked to the sugar present in our food. And I go to the fourth one, the industrial products. So the ion exchange, chromatography, the technologies we have can be also applied in the chemistry industry, such as like specialty chemicals, phosphoric acid. Since the past few years, we are pretty big on lithium purification. But there is also fermentation, and that's obviously the most important topic today. For us, the big boom we saw was in the years 2000-ish in Asia, where the amino acid production was really exploding. Amino acid and antibiotics, and all of that is made by fermentation. And at the time, it's very interesting to see from one market to another how people deal with the challenges they have at R&D first, and then at industrial scale. But in Asia, it's pretty well, I would say, acknowledged that the clarification of a fermentation broth shall be made by using ceramic filtration instead of what's typically the case in America, centrifuge. A very interesting case right now. I'm sure a lot of you guys have heard about Liberation Labs, the very famous CMO company that is about to open, or maybe they are already open. They made some very interesting case studies comparing different DSP technologies for this pretty important step, that is the clarification of the broth at the end of fermentation. And the CEO himself said that without using ceramic filtration membrane, it would be difficult for a bio-based industrial company to actually make money because of all the advantage you get by using this technology. So that's basically how we really developed a lot the technology in Asia in these years. And you'll see later, it doesn't stop here, because thankfully, some of the bio projects are pretty quote unquote easy, if I can say so. So some people are gonna be infuriated to hear that, but I would say some projects are easier than others. I mean, typically, if you can easily separate your biomass from the product of interest, if the production of the product of interest is made externally and not internally, depending on the viscosity of your biomass, all of this, a lot of different parameters are going to enter into account here and help you with the type of technology. So about the company, really, these are the markets that we are addressing. I already mentioned it a little bit, but our core technologies are so ion exchange. That's where everything started. We are also using chromatography, which is also a resin-based technologies like ion exchange. The difference being that with ion exchange, you really exchange ions, either to catch an impurity or to catch the product of interest. It's the case, for example, for a famous amino acid, lysine. We do that. We catch the lysine on the ion exchange and let the impurities go through the bed. While chromatography is more relying on other properties that make that the interaction between the product and the resin make that within the product, some molecules tend to be retained, not caught, just retained or slowed down, I would say, while some will go faster. And at the end of the column, what you collect, and if you collect at different times, then you will be able to make your purification. So same type of consumable inside the column, but different kind of a strategy for making your purification. The other one, the third pillar of the technologies that we supply is membrane filtration. For bio-based projects, I love the combination of ceramic for really separating the biomass from the supernatant for what's outside of the cells, combined with typically polymeric membranes that are a great tool, a cheaper tool once you have something that is cleaner to do a separation based on weight. We also combine these technologies with others, such as catalysis, enzymes, electrodialysis, active carbon, etc. All of that to get to the final product. The idea behind the combination of these technologies is that by combining the technologies, it's very difficult to do everything in one step. So by combining the technologies, you have the capability to work on different aspects, to work on different strategies in order to get there, in order to get your purity. Now we are Applexion, we are beyond purification. When we were still part of Novus Group, it was when purity is key. So all is about purification and getting there, getting your points of purity. The way we work typically is we see it as a wheel. Because once we have installed equipment, the story doesn't stop here. So typically we start here, depending on the project, but with most of you guys that are on this call today, it starts with R&D, bench scale, going to piloting, then going to a system, an industrial style system. The offer comes with digital tools. I'm going to talk a bit further about that help you on logistics point, also on getting better performances, on understanding better how your system is working. And once your system is getting old, because we've been here for 60 years, so we have a lot of clients that installed units already a long time ago. Then you always have new demands. I want to consume less water. I want to consume less chemicals. I want to generate less effluents. I need to work on a new product. All of that can lead to what we call revamping projects. So for the first part of the wheel, basically it's the R&D part of the project. As I was mentioning before, our main technologies are cross-flow filtration, chromatography, ion exchange, and we can really cover a pretty wide range of steps according to where you are in your process. So obviously it starts at labs, but we also deal with people who already did a lot of the work at lab scale. They need to go to pilot straight away, to start to produce a little bit, to show the potential investors the final product, that it's concrete and that they can already do some other additional tests on it. We can also do some tool manufacturing, etc., etc. So scaling up, and I don't want to get too much away from the topic of today, which is scale up, is passing by piloting. There is a lot of the work is done at bench. You very often for all of the chromatographs, for all of the technologies I mentioned, we can already get a lot of information at bench scale. It's giving us really some good ideas of how the system is going to perform, how big is going to be the system, what are going to be the operating costs, how much water, chemicals you're going to need, this kind of thing. But between the bench and the industrial scale, there is a must-go phase, which is piloting. So we have a fleet of pilots that are basically traveling the world, either for ion exchange, for chromatography, for membrane filtration. And we want to be flexible, especially with fermentation projects where you make your fermentation, you're not gonna load a tote with a cubic meter of broth, and then it's going to travel who knows where, and then you're going to apply the DSP. It's most of the time you cannot do that. So we try to either work at people's site and send our pilots on site and people to run the pilot, or and to train also the people on site to use the pilots by themselves. We also have some partnerships with some CMO companies that can work at pilot scale, and then we can do that. You have some options, more and more options, like our partnership with the IBRL that is looking like a one-stop shop for this kind of thing, where they can do the fermentation, and we have our pilots over there on site. So it's also a great solution to do everything together. So every project has its own, I would say, parameters, challenges, and we try to be flexible with that necessary piloting phase, which is helping you to do the scale up. So typically, you start from the R&D, you do your process design, and then once we are done with all the R&D and the piloting, then we can go to the engineering phase. We have different teams of engineers over the world, going all the way then to finally commissioning and install industrial equipment. Here is actually a nice picture of it's a bio-based plant. It's in China, if I recall well, and this unit is actually used to clarify fermentation broth at the end of the fermentation. One other aspect that is important when you talk DSP is the consumable, because a lot of the DSP technologies are gonna use consumables to run. Typically on ion exchange or chromatography, it's gonna be the resin that you're gonna have to change from time to time. And for filtration steps, it's going to be the membranes. Either they are ceramic or polymeric. So we also have specific services for after a certain time when your project is installed and running, and you need someone to have a look and check if the consumable needs to be replaced or not. Coming back on our digital services, I would say this is really a part of what we supply that has been exploding in the past few years, and it's really linked to the. You can make the parallel with what's really happening right now. We are having a conference on Zoom. Probably 5 years ago, it would not have happened. It would be too complicated. Skype was not working, etc., etc. But the COVID made that suddenly there was a full acceptance on the technology. Something that happened with COVID is that a lot of people left their site, and a lot of industries, a lot of factories are suffering from the lack of people with know-how. And I'm sure you, like me, you have seen a lot of these articles. Where have been the people? And we see that a lot in the factories. And we had a lot more and more demand of how can we run things. And so that's how we came with a set of tools. One being a Run 4.0 that is really like a doctor. It's a health check. It's basically we saw a lot of very interesting presentations yesterday about digital tools. And I remember this one, I think it was Ganymede that was showing these dashboards. So we do that but for the industrial environment. And the idea is to generate dashboards where you will have a health check, and the operators, even operators who are not who are pretty new to the factory, can easily receive feedback and see, okay, here there is something wrong. This is what I should check. Now the second one is perform. Perform is really about when you install an equipment and you train people, and you realize 10 years later that half or 75% of the people you trained are not here anymore. If the knowledge was not passed properly, people don't know how to get to the best of their units. And we are installing Perform in more and more places. There is an incredible case study in France where we installed Perform because they were not even realizing that the unit was not performing very well. And they made, by installing Perform and by having, I would say, an autopilot that is telling them exactly what to do to get the best performances, they save millions of USD every year. They didn't even know they could do that. The third one is Link, and it's linked to all the handling of the consumable. So it's a system integrated with your phone, and you have QR codes on the bags of resins and membranes, etc., to be sure that you know where is what, when what has been changed, and when it needs to be changed. The revamping, it's the last part of the loop I presented you. We are implementing on older units a lot of with our after-sales service. We are implementing a lot of revamping missions to help them make to get to better performances, and also there is a lot of incentive to get better environment, I would say, impact, or less worse, if I could say so. One being on chromatography systems, to help to install a system that allows the chromatography to never stop. One being a recycle boost, which is on either ion exchange or membrane, to install loops within the system so that you can reuse chemicals and drastically reduce the consumption daily. And the third one being dedicated to ceramic filtration, and to also help decreasing the electricity consumption. So yeah, part of the expertise we have is really to help the people, and so we can do technical audit, go on site. We have I'm part of we have a committee inside the company called the Decarbonation Committee. So we are trying to really work hard on lowering the impact of our equipment on the environment. So we can really help you to work on assessing what's your impact. We also have a pretty good team on everything that is linked to intellectual property, which is a very important topic that we have at very early stages. We can do training, and we can do a remote assistance, either it's on site or remote. Talking a bit more about the bio-based projects. This is basically, I presented this, it's your dream. Your dream is a fermenter. You have your bugs that is nice and growing well. You put a bit of nutrients, and then you get a nice and clean final product, and it's all good to go. But that's not the reality. The reality for bio-based projects looks more like that, more like a fall. It's a mess, and there is this war that will never stop between the USP guys and the DSP guys, and we never know whose fault is that. But I've been working too long. I've been a bit in the USP, but I've been too long in the DSP. So now I'm a DSP guy. So the fault is on the USP team. The reality is that you have a lot of things to remove to get your final product of interest. And you want to remove minerals, suspended solids, fats, cells debris, sugars, protein, etc., etc. These broths are very, very complex most of the time. So the way we see it is the DSP is a one-stop shop. Most of the time won't happen. If you are lucky on some food application, you have some chances with some pH change. You can have a protein that is changing its form, and then you can separate it pretty easily and then go on evaporation drying. But most of the time, for more complex molecules, you'll have no chance. You'll have no other. You will have to go through several steps. The way we see it is so a pre-treatment. The first one being with a ceramic filtration to get a nice, clean permeate that can then be treated on further steps. if needed, especially according to the distribution of molecules remaining at the outlet of these membranes. You can use polymeric membrane with a tight cutoff to do a separation by size. And then, according to what you have, what's the impurity you have, what's the final purity you need to reach, and what's the impurities that you are trying to get rid of, you can work with chromatography and or ion exchange to get there. You very often have, most of the case, you have a final step, either to remove color or to concentrate by evaporation, crystallize or dry sometimes, depending on the form of the product you want to have. So about the scale up again, for us, it's really about taking the steps one at a time, and we like to work with gates because it can happen, unfortunately, that at bench scale you realize it's not going to work. So what we are doing is that we get together with our partners in the bio-based industry, and we work on the feed material. We analyze it. We look at what kind of pretreatment we can do, and we do a bench scale. And from there we already from the trials we make at bench scale, we run our in-house simulation tool. And basically from there, we can do a first process evaluation. We can already know, as I was mentioning before, more or less where we're going to stand instead of expected performances and in terms of size and cost of equipment. In CapEx and OpEx, obviously, because it's both I consider that both as almost as important one to another. If it works, if the numbers look good, then we can go to pilot scale. And then we run additional trials at pilot scale. We can start producing samples, etc., etc. And the pilot scale will allow us to confirm what we've been simulating at bench scale. From there, we can make an offer for an industrial system with process performance guarantees on the operating cost, on the purity, on the recovery you're gonna have. So we go to process design, industrial project, and finally we commission the equipment. So the idea of working step by step is really to test and de-risk your purification process. There is no point starting at piloting. It's going to take time and money if at bench we already see that it's not going to work. It's also working by step like that allows us to benchmark the different solutions that are available. It's helping to transition quickly from early stage projects to industrial applications. And then, of course, we're working with people like all you guys to produce new food, functional ingredients, and bio-based materials. So that's it for my presentation. We do have a few minutes if there is any question, right, Ning?
Yes, thank you, Antoine. You have a question that let me read out. So the first question is, does your membrane separation design allow for fine-tuning pore sizes or shapes for specific compounds of interest?
Yeah, absolutely. So for the ceramic membrane is, you can imagine that for the ones who are not familiar with the technology, is basically a porous stone that has a cutoff. And the finest we have is 15 to 50 kD, so 15 to 50 kilodalton, and up to 0.4 micrometer. Typically, the star of the ceramic filtration for clarifying fermentation broth, which is really what most of the people on the call here are looking at, is between 300 kD and 0.1 micron. And that's allowing you to do most of the clarification. Now that's for the porosity, and now and the other one is the shape. It's the person who asked the question probably knows about that. The membrane, the channels by which the fluid is going can have different types of shape. And the idea behind is that you have some fluids that are more viscous than others. Some shapes are more adapted to more viscous broth. The drawback is that these shapes have less filtration area per membrane. So it means that at industrial scale, if you need that membrane, you would need more membranes. So it would be a bit more expensive. So one of the key things when you do scale-up trials for broth clarification is to, at this stage, you're not anymore looking at the porosity. You already discovered that at bench scale. But at pilot scale, when you do the scale up, then that's when you identify the perfect shape for your project. You want to find the shape that has the highest amount of filtration area per membrane, basically the best adapted shape.
All right, thank you. The next question is it feasible to replace the centrifuge with a ceramic membrane filtration or microfiltration for commercial scale production?
Absolutely. And that's a big demand for us. The demand is exploding. I mean, I think centrifuge is very good for some type of application. The problem is that the US market has been taught that the centrifuge was for everything. And I personally think it's wrong. And that when you see where Liberation Labs is going, that's an absolutely proof that on a lot of different applications, the quality of the permeate you want to have requires actually to have ceramic filtration. So yeah, we are replacing in a lot of factories centrifuge by ceramic filtration.
Great. All right, I don't see other questions. Thank you again, Antoine, for the great presentation. If you have more questions, please put it in the chat. Antoine, maybe you can move to the chat and just to answer extra questions.
Absolutely. Thank you, Ning. Thank you, everybody.
So let's move to the next presentation. We have Morten Christensen from GIA, and Morten will talk about precision fermentation DSP. Okay, take it away, Morten.
Thank you very much. I hope you can hear me.
Yeah.
All right. Yeah. Well, welcome everyone, and thanks for joining this session here. My name is Morten Christensen, and I'm the application manager for biotechnology at GIA. For those of you who do not know GIA, GIA is a equipment and solution supplier for well, biotech industry, but also food and pharma and chemistry, etc. So we do a lot of engineering work and provide these turnkey solutions or individual unit operations. We're very big in precision fermentation these days, but in this presentation here, we're only really going to look at the downstream process. So what I have planned for you today, if I can get this to Swift, is now first we're going to look a bit on the strategy side. So how do you develop a downstream process for your target application? And then we're going to talk about the processes afterwards that you need in order to achieve these different steps in your strategy. And finally, we're going to talk a bit about KPIs. So which KPIs in the downstream, but also on the fermentation side, has impact on the equipment, and how does CapEx look? So what should you expect in terms of how much does the downstream equipment cost compared to everything else? But first off, we're just going to look at how to then develop a downstream strategy. So of course, the downstream is straight after the fermentation. So we're going to start at a point where we are at the end of fermentation, and now we have a reactor tank, and it's of course filled with the broth, including organisms and non-spent media and process additives such as antifoam, etc. And now we have to figure out how do we then clarify this. So the first part of the downstream process is usually referred to as the recovery session. So this is where you try to remove the biomass and technically the GMO component from your product, because you need a non-GMO product, and these organisms are by definition genetically modified. If you like, I have depicted here, have an intracellular product. In this case, it actually makes sense to start out by removing everything that is not the cells, because that's an easy task. and not so much the media maybe, but if there are metabolites, so other proteins that's been excreted, there will be some cell debris, etc. But there will also be antifoam in this broth, and it's very nice to get rid of early on. Especially the antifoam can really screw up the later purification process if you have filtration units. So it's very good to get off in the recovery process. Exactly how to achieve this, I'm going to go into in the next segment. So for now, we're just talking about the strategy here. As soon as we've done this, you now have a washed biomass. And of course, we then have to extract our protein of interest. Usually it's a protein. It could, of course, be another biomolecule. And of course, we do this by breaking open the cells. This can be done chemically or with enzymes, so biochemically, but we prefer to do it mechanically, just so you don't add something to your now washed cell biomass. So we usually use very small hammers. We use homogenizers for this. Going to talk about those in a few minutes. But you need to understand that as soon as we do this, you're working a bit on the clock, because all hell sort of breaks loose as soon as you lyse the cells. Unless you're working with bacteria, all eukaryotic cells are pretty compartmentalized, and as soon as you lyse them, well, everything mixes. That includes proteases and other enzymes that would be inside these cells, but also other components that can make either your target product or other organic compounds. They could start to aggregate or precipitate. So there's a lot of complexion that happens if you don't work relatively fast after having lysed the cell membranes. So what you want to achieve now is to remove the cell debris, so membranes, organelles, and also the DNA, RNA part of the cells. So we're separating that away. And now we have a pretty clean broth in terms of cell material, and we can call this GMO free. We have a GMO product, and now we are then out of recovery. So that would be the recovery part for an intercellular product. But to be honest, especially within new food where we produce a lot of proteins, it will very rarely be an intercellular product. It will most likely be an extracellular product in order to get the right post-translational modifications. And that changes a bit how the recovery looks. So you can see here depicted the broth is now very different just because you have the proteins and other metabolites that the cell will excrete. And we can't really start out now by washing the biomass because there's it doesn't make any sense. But we can start out by removing the biomass and cell debris, and that is what we're going to do. So you remove this in a few unit operations, and usually you also make sure that you remove the antifoam again, like I told you before. So this allows you to end up in fewer process steps that with intercellular products, you end up with roughly the same type of stream entering into then the purification part. Now, recovery is usually quite samey. It's the same type of unit operations every time. They change a bit, of course, from organism to organism and product to product. But purification is far more application specific, because what you need to do now depends a lot on what you want to use this product for. So especially in the food industry now, if you're producing a lot of whey protein with precision fermentation, and it's for, say, a cheese type or something like this, you might not need to purify it any further. If there's amino acids and some salts and maybe a few other proteins inside, it's not a big issue, because if it's for cheese, well, the color and the texture and the taste will maybe not be very greatly impacted. So you could maybe just concentrate, in this case, the broth coming from recovery, and then go on to the further post-processing. If you have another application such as, say, ice cream, well, now suddenly taste and color might be impacted by the smaller molecules, especially if they're coloring agents like vitamins. Well, in that case, you might need to do some excessive washing. in order to get rid of these, so that you have a more pure biomass, not biomass, protein concentrate at the end of purification. If you have something very strict like infant formula, you might not even be allowed to have other proteins in. And in this case, you need quite specifically to either pick out your target product, or most likely you're going to try to pick out the contaminating metabolites. So that would be the other proteins, simply because they're supposed to be in much lower concentration than your target protein. But if you have a vast array of other proteins, it might be a tough ask to pick it out. So that would be the strategy that you would typically try to achieve in a downstream process. So the technologies, on the other hand, that you need to use are tried to depict here. So of course we have the expression, then we have recovery and purification, and then we have a post-process, which is typically a drying step. So the first thing that happens after we have had expression and we need to harvest is we need to stabilize the broth. And typically this is done with some sort of heat treatment. So a lot of the time this is just a cooling, because it will be an extracellular product, so we don't want to lyse the cells. So it could just be a cooling heat exchanger, but it could also be, as depicted here, a pasteurizer. If you have an intercellular product, you can pasteurize it, or in some cases you can also do it for extracellular products, and to completely inactivate the organisms. So that is two different approaches. Usually you will see that it will be a broth cooling that occurs at this point in time. Again, coming back to the intracellular products, I mentioned that we will be using homogenizers in order to break open the cells. This would be a typical industrial homogenizer unit. It is quite efficient. If you can go up to around 1500 bars in a single pass, and usually you have several passes if you have a very hardy organism. The only thing you really have to take care of is the temperature. For every 100 bar that you have in a homogenizer, you're roughly increasing the temperature with 2.5 degrees. So you can imagine even going to 1000 bar in a few steps. You don't have to do many steps before you are reaching a temperature where your product could denature, if it's a protein, or even if you don't denature anything, at least you are creating conditions where something can grow in, right? Because you would be increasing to 30, 40 degrees very rapidly. So you need to maintain a cool broth whenever you do homogenization. So you need cooling as well. Then when you have your broth with cell debris or cells and product in solution, we of course have to clarify it. And there was already a good presentation on it previously here, talking about ceramic microfiltration. This is also, I think, the standard solution in GEA. It's a super flexible system, and it's a hard border for your GMO. So it has a very narrow cutoff, and in many countries it's sufficient for as a hard border towards the GMO. Sometimes you need, on the permeate stream, you need a dead end filtration, microfiltration, in order to claim that it is GMO free, to making sure that you remove all DNA traces. It's very tough to prove that you're removing all DNA traces, because we're not working aseptically in the downstream of a precision fermentation. So it will be contaminated by other organisms, which are of course spreading their DNA as well. So there's it's quite tough to prove, but of course we have to do our dandiest to make sure that the organism doesn't go further downstream than the recovery process. The ceramic microfiltration system is also nice that it removes antifoam, and it can get cleaned afterwards. So it's much preferable to remove the antifoam in this step compared to later when you get to ultrafiltrations or similar, which is typically not ceramic systems when we are in the purification area. Not all organisms can go directly on microfiltration though. Sometimes we need to have separators in place. They are very rarely a replacement for the microfiltration, because they are not a hard border. So if you have a food industry or something like this, you would not just have separators in. In other applications, you could see only separators, but there's almost always a microfiltration in. But if you have a fungi or a yeast type, it's usually preferable to have some sort of separator before your microfiltration system in order to achieve the high uptimes before fouling completely takes over the system. So typically that would be a disc stack centrifuge, or it could be a decanter centrifuge, as shown here. The decanter is really only for large organisms or large structures. That would be mycelium forming fungi or similar, where you would see these decanter systems. Otherwise, it's preferably these disc stack separators, usually nozzle separators, because you have very high biomass concentrations in these precision fermentations. And it's also very rarely just one system. It might be one system if you're at pilot scale, but as soon as you move to production scales, you very typically have a few in series. And this is in order to achieve, of course, the correct yield and the correct capacity, along with the correct water content, because you're using washing in these steps as well in order to achieve the right yields. It's very hard to just do a concentration with very little water input without having a very poor yield in the separator system. So depending on, if you don't do this engineering correct, you'll end up with adding way too much water, and we're talking about maybe 2 to 3 times the amount of water that you have in your reactor system that you are adding in the downstream during both the separator part, but also the microfiltration. And that's a lot of water. It's a big hydraulic load to move to the purification. So you need to minimize this. And also you need to make sure that the biomass waste stream that comes out is as concentrated as possible, because this is something you have to pay in order to get rid of. So that is another big benefit of separator systems, is that usually the concentrate, the biomass comes out more concentrated than on the microfiltration systems. So that makes that whole later handling of the biomass side stream more manageable. Now we're out of recovery, so we passed the microfiltration system, and now we have to then wash and concentrate the product stream. And this we usually use filtration units for, so spiral wound ultrafiltration and nanofiltration systems, or sometimes we use evaporators as well. These are very good for concentrating in some applications. And we can also, if you have a whey protein concentrator, are usually performed with membrane filtrations alone, so you can get to these 30+% of protein in just a nanofiltration system. So this is good enough, like I said, for cheese applications. Sometimes you need to do something that's even more specific, and then we're moving into the applications that are very expensive to get on a high capacity. So we're talking about these, say, active carbon columns, for example, in order to remove coloring constituent, or we could be talking about chromatography in order to pick out either other proteins or your protein of interest. If you're picking out your protein of interest, you might have an issue, because of course in precision fermentations for novel food, at least, or new food, the titers are very high, so your columns will be saturated quite fast. And also you have to release it again, and usually you release this with a brine, so you also have to run a desalination process step afterwards. So there are some add-on effects by doing this. Another approach is, of course, to still use the membrane systems and then do a precipitation instead. So if you can precipitate either your target product, or if you can precipitate the contaminant, for Pichia pastoris, for example, it's a quite typical example that mannose would be a contaminant in high concentrations, and then you can precipitate your target product, remove the mannose, and then re-elute your target protein. That's one solution. I haven't mentioned yet that you also usually need to take care of the bioburden throughout the recovery and purification part. So as soon as we're in purification and we have our very clean product at the end of it, we might need to reduce bioburden because we are working hygienically, not aseptically, before we go into post-processing. So where you're typically drying or maybe filling your product. In that case, we might need to do either a microfiltration or it could be a pasteurization in order to reduce the bioburden before it's a final product. All right. So finally, here we're going to talk about KPIs and the CAPEX of the system. So I'm going to show this overview again and talk a bit out from here, because it technically follows the errors that if you have a KPI going wrong in recovery, it really just impacts the other equipment in recovery as well as purification and further post-processing. So we have to take care primarily about what we're doing in the bioreactor, because that really impacts all the way down. And that, of course, is whenever we're designing a downstream process, it depends a lot on not only the size of the reactor and how many reactors do you have, but also the mode of operation. Because if you're running in a fed batch system, a lot of the time you have these long turnaround times. So say 3 to 5 days. And that means that you need 3 to 5 reactors in order to have 1 reactor per day that you can harvest. And then you're harvesting 1 reactor and you know roughly what to do with your downstream process in terms of capacity, how much it should be able to handle. If you then do strain development or something similar and you can suddenly run continuous on your system, the downstream process will not be suitable at all anymore. Because if you're running continuous on your reactor, suddenly the stream that comes out is very small compared to when you're just harvesting an entire reactor. So it's very important to focus in early on what will be the mode of operation on my reactor system. If you can have a very good perfusion reactor type, so you can retain the organism and just extract your protein of interest, well, you can have quite a high capacity system if you plan for it. But it's very hard to do if everything is planned for a fed batch system initially. Another thing, of course, what everybody talks about whenever we are talking about bioreactors is the titers that they can achieve. So the titer of their given strain. And it's, of course, very important, especially for the recovery part. So both the titer of the biomass, how much do we have to remove, but also just the concentration that the product comes in at, because, of course, that is less water and other things that you have to remove in the recovery step. There's, of course, the purity as well. It impacts more on the purification part, because you might have an organism that is developed for super high titers, but it's no use if it's also a very impure broth that comes out. So this was also mentioned in the previous presentation, right? That this is the nightmare for the DSP, is that something comes out that's a pure soup of different organic material that you have a very hard time actually handling in the purification part. So all of these KPIs play together, and they impact a lot on how expensive the DSP becomes. But how much does it cost? How much does it cost if you compare all of the unit operations? So not the entire factory, but just the unit operations that you need, all the way from media preparation to powder handling after the drying. How big a part is the DSP? Very typically, and please understand that these numbers vary a lot. So this is an average number, but very typically the downstream is costing a little more than the upstream. So in the range here of around 55% is the downstream. That includes the drying in this case. So I have a further breakdown you can see. So typically you have a fermentation, which is costing around 35% of the total cost for the unit operations. And then, of course, you have around 30% for recovery and purification. If you're going with something for infant formula, expect this to explode quite a lot. You can have significantly more than 55% for the downstream process. You can you can easily go to the 70s if you need something for infant formula. But it can also be reduced quite drastically if you have quite a pure production in your fermentation and you need it for something like cheese. Well, you might only really need a UF or an NF in the purification, and then you're ready to dry the product. So it depends a lot on the application, what the cost will be, but this is not too uncommon that you are in this range of 30% for recovery and purification. And so it's a big part of your overall investment. And it added to this, of course, comes all of the support equipment that you need. So that is, if you have a lot of washing or if you have chromatography or something like this, well, there's all of the preparation part and handling of wastewater, etc., which comes primarily from downstream process, and that you still need to that equipment you still need to take into account. That's not accounted for here. This is the price for the equipment, the direct process equipment. So I really hope that was of interest to you. Let me know if you have any questions or comments that you want, and you can reach out afterwards. You can also go to GEA.com, of course, and find your local contact. Thanks for listening.
Thank you, Morten, for the great overview of the downstream processing. I don't see a question in the chat yet. So I have a question for you. What do you see the main challenge when you develop a downstream process at scale?
Yeah, I think a challenge a lot of the time is that that balance between the capacity of the unit along with the amount that you need to wash in order to achieve the right yield. So I think that's, we usually would aim for a unit operation to have less than 1% loss in a unit operation, because it very quickly adds up. But it's actually very hard to achieve without having suddenly too low capacity, because you are excess washing with water, and of course all of this water is just lowering the capacity of every subsequent unit operation. So a lot of the time this needs a very big optimization effort after the plant is installed. But I think this is something that is usually it's not considered to the necessary extent when we, especially when we talk to startups. They quite often have their fermentation nailed, so they know exactly what they would want to do with the fermentation, but then they have a vague idea about the downstream process, which is just as important as the fermentation, to be honest, because it just impacts your business case. Completely ruined if you're losing 25% of your product in the downstream process. And like I've just shown, the cost of the equipment is very significant. It's around the same order as what you spend on the reactors themselves. So it's important.
Yeah, absolutely. Also, what's among the DSP units operations, which one is the most frequently used based on your experience?
Yeah, there are a few usual suspects, I would think. Again, it depends a bit on the application, but for precision fermentation, I think the ceramic microfiltration is almost always in, just because it is a hard border, so it's good for a lot of legislation. I think primarily here for new food, and you will almost always see a ceramic microfiltration system, or well, it could be hollow fiber or something other than that. But we have the ceramic systems. We're very happy with those, and they do have the very nice cutoff for the antifoam as well, which is just important. So, but in some industries you can have just separator systems and no microfiltration. It's very rare for food, I would think. For new foods, there's almost always a filtration system in.
Yeah, great. Yeah, that's also aligned with our previous speaker.
Yeah.
And there's a ceramic membrane filtration. Also, there's a polymeric membrane. Can you talk about like what's the main difference between these two?
Yeah, yes. So of course I'm not one of our absolute experts on filtration systems, but I can talk to it. So if you have a ceramic filtration system, the pores are usually far more defined. So you have a what's it called, narrow or cutoff, And the system can handle far more harsh conditions. So you can clean them with very different settings and cleaning agents than you can the polymeric systems. So the polymeric systems, of course, have the advantage of capacity and the size that you can do these units with. So they are usually what's used for the purification part, where you have removed the biomass, you have removed the antifoams, and you're really working with proteins and sugars and stuff like this. Then for the UFs and NFs, they are typically in the purification part, they are typically polymeric systems.
Great, thank you. We do have a question in the chat now. Do you have any suggestion for simultaneous separation of protein and fatty acids from the biomass? Could they merge into a single downstream process?
Oh, that is tough. Well, only it depends on your target product, I would assume here. Unless the fatty acid is also a product, I don't assume so. But if you want to remove it, if your target product is much smaller, then of course you have the option of just going directly to a pretty narrow membrane. Otherwise, there's very little in common for the proteins and the fatty acids that you can separate them based on, I think. So no, I have not seen it, and I cannot come up with something just on the top of my head.
All right. Thank you.
But thanks for the question. That was good.
Yeah, if you have any follow-up question, please feel free to put it in the chat. And Morten, maybe you can stick around and take a look if there's extra questions. All right, let's move to our next speaker from FermenteQ Innovations. Do we have Sri? Great. Hi, Srinivisa. Hopefully I pronounced your name correctly. And the topic for this talk will be on advanced bioprocessing and scale-up equipment. Please take it away.
Thank you very much, Ning, and then Deepti for accommodating in amazing Expo, which gives a kind of an interactive with competitive and then collaborative companies. I'm Srinivasa Reddy. Just before going into the presentations, CEO of FermenteQ and then ProEntech. We are over a decade experience in the previously known for the different name of the company, established in Asia, India, and then provided services around India and then also in the Middle East. And then in 2023, we renamed the company towards the global adaptation. So for FermenteQ and then ProEntech. So what we do here is a tech family. I am more fascinated toward the Q. Q is a quantum energy. That's why I carry forward the Q in the entire my companies. FermenteQ, ProEntech, and then IVTech. What it does is kind of providing the biological bioprocessing solutions and the non-biological chemical processings and then beyond bioprocessing towards the different scalabilities. That's where we form the different companies, and then we have over 750 members in the total, all the three companies working. So here we are bringing up a kind of an expertise in different groups here. One is in the AI and the ML from the IVTech, and also vision integrations. VI is the vision integration into the bioprocessing and the processing industry, because most of the processing industry is going in operational efficiency, enhancing efficiency of the scale up. So we need to have a much more efficient in performing the job. And then a second thing, FermenteQ, we are focused on producing the lab scale, pilot scale, and then commercial scale. Commercial scale is over 200,000 liters capacities. We are providing solutions. That's what we are. Particularly talking about the FermenteQ, I'll talk about a little bit what we do is we do the custom designing and then whether it's a complete bioprocessing systems, solutions, or turnkey solutions. In this process, we don't do complete end-to-end solutions, the manufacturing of equipment. we collaborate with companies which is validated into the DSPs and also some other critical technologies where we can able to integrate. We do have our own internal manufacturing. Over 60% of equipment from the lab scale, again, like to the completely over 200,000 liters. Our strategy is a fit for use kind of things, like a purpose built, which we can use, whether it's a fluid dynamic, pressure controls, and then IoT integrations, and then advanced bioprocessing integrations. That's the team that what we have, independent experts over 25 years of, at least 25 years of experience in each individual's having from the previous life. So that's where we are. We don't say the completely unique, because we carry forward at least 10% of the uniqueness comparing to the global companies. The other turnkey industries, we are industry focused, and then flexibility supporting in independent productions, because every section of the industries has its own uniqueness. For example, the private companies or independent companies productions that has a uniqueness in producing the single candidate of selection, or again is the CDMO. So CDMOs has a multi-candidates, whether it's prokaryotes or eukaryotes or anaerobic or aerobic. So dynamic process creations is a customization, a responsibility of a customizations. So we focus completely towards how we can able to accommodate the system, whether it's an internal environment or the outside environmental integration. That's what we discuss with our collaborating clients, and then work with the long-term relations. We have our global compliance that can need the ASME or a TUVs or a CEO, CE or ISO certifications. We have in place to provide it. And then we have a diverse application solutions, whether it's in a biological sector, whether it's in we don't manufacture single-use reactors, but we have capabilities of collaborating with other companies. But we do have the non-human vaccine production capacities, and then microbial prokaryotes and the eukaryotes, non-microbial as well. The sustainability is our focus, that keeping an energy efficiency are our the ETP plans, that is extremely valuable for us, how we can able to reuse the media. So the technology, that's where we are looking forward to collaborations with our filtration technologies, and then other partners. So why we do the scalable technologies? Is there a necessity? Yes, I think my previous companies, most of them projected the same thing, whether it's TetraPak, very well articulated, or they projected a growth of the population by 2030 and then 2050. It's extremely complex to provide the food, and then also the bio-based materials, whether it's a synthetic or a biosynthetic. Conversion from synthetic to biosynthetic is already started. That's where the bioeconomy is growing, addressing over $3 trillion by 2030, and then $30 trillion by 2050. That's where we can able to contribute what we can able to be uniqueness comparing to the existing companies. That's where the advancement comes. So to address the global bioeconomy, or what are the industries that we focus? It's every industry that comes into the producing, whether it's a proteins or cosmetics, or whether it's textile industry, colorings. It's wide varieties of the industries, whether it's in pharmaceuticals, nutraceuticals, biotechnology companies. We work with a wide range of industries. How our process starts? Our process starts with a URS, and then it goes with a complete assessment, and then providing the DQ, and then ongoing complete annual maintenance contracts. We believe in a long-term the relations with our companies, because scaling up is a challenge, and then from the pilot scale towards the large scale, if you wanted to bring the companies, the processing, you need to have the manufacturers associations in the long term as well, because the manufacturers knows some of the critical technology operations. So that's where we believe in long-term relations with our clients. We don't provide just as equipment, we provide a solutions. We believe in providing a solutions is the best possible things that, again, as mentioned, we produce over 60% of the equipment. That equipment includes bioreactors, fermenters, a filtration system, whether it's a mobile reactors or a skid-mounted reactor, from the pilot scale to the large scale. We cannot make it the mobile reactor, integrated CIP system or a hybrid CIP systems. We have our own in-house capabilities to design it custom, or also it's a single skid dual reactors, something which is very unique for the pilot scales. So we can able to make it everything from the scrap, like from the sheet to complete performing reactors, we can able to provide it. So how our why FermenteQ? FermenteQ is we take consideration of a process when we talk to the client. We do the background research and development. Once we have done, we proceed with the custom designing. And then once we are done, whether it meets the global quality and then compliance, that is our primary the thoughts that we would like to compete and then provide a solutions. A scalability, the based on the compliance and then custom designing, the scalability, it is extremely important, like how we integrate the components or required technologies. We have a very well articulated teams, and then we can segregate the production line, and we are completely competent on lead time productions. And then the validation, in-house validations with the FAT, and also providing a proof of every the FAT documentations. Then we send it to the clients, and then in installation and commissioning, and then we also look forward to long-term collaborations. So what type of organisms we use? I think it's a wide varieties of organisms, whether it's a biological or non-biological. We use it, we designed for the equipment. Again, as mentioned, our team is capable of handling wide varieties of producing bioreactors, fermenters, aerobic, anaerobic, and various sectors. That we do have a advanced automation bioprocessings. Where I'm not completely detailed discussing here. Anybody is interested can contact me. We have a separate segment for the processing integrations. Whether it's in automation, whether it's a fully automated, semi-automated, and then advanced automation systems. We integrate AI and ML is. It's already in the line. We design along with our customers' needs. How we are distributed all over the world, we work with the strategic collaborations with our partners, whether it's a clients or not necessarily with the clients, even the service providers, companies, they have the technologies that what we don't have. We collaborate with it, especially DSP. We look forward to collaborating with a lot of different companies in the different countries, just to make sure the cost efficiency we can able to provide it to the client, and also enhancing the quality, and then also scale up. Scale up is like a commercialization. Scale up is we need a perfect a precise expertise into the system. So we collaborate. We are ready to partner with companies. Our portfolio includes biologicals and non-biologicals, and then when we talk about, we definitely have a portfolio discussions on an NDA, or in the scale of like 1 liter to or 150,000 liters capacities. That what we have provided, whether it's in ethanol productions, which is non-biological side, it's more like a enzymatic and the chemical processing, and then the biological processing as well, protein productions. In summary, like what we do is we have innovation and R&D and the lead time, the leading trends we put forward, customization and fit for use, scalable to the over 200,000 liters capacities that we manufacture, and quality complaints, and the lead time is something, and the cost efficiency. When in the name of customizations, there is a lot of assumptions that we need to add extremely complex equipments. And then globally, when we see the pharmaceutical difference between the pharmaceutical the sector industries and then the biotechnology, non-pharmaceutical sector, it is more weighing in a non-pharmaceutical sector. So it's not necessarily to have a highly decorated systems. So that's where we can focus on a cost efficiency, providing kind of a partnership that can enhance, whether it's in SMEs, MSMEs. They need kind of a partnership that can go forward and the cost efficiency so that they can prove their validations and then a concept to the commercialization. So that's where we are strongly aligned with a vision to integrate with a global service provider. We also have our equipment partnership where we do the kind of an investment strategies to support the SMEs and then MSMEs. That's where we are coming into the supporting the scale up. Yeah, that's in a quick. I would like to keep it as fast as possible. Yeah, thank you very much. If any questions, I'll take further discussions.
Thank you, Srinivasa, for the great presentation. This is a 30-minute slot, so we do have 15 more minutes for discussion and questions. I don't see a question in chat yet. So I'm wondering, what's your approach? I know, so your company work on both upstream and downstream. So I'm wondering what's your thoughts on process integration of the upstream and downstream processes?
When it comes to the upstream, we manufacture our own equipment. Downstream, we don't manufacture entire systems because it involves a lot of variabilities, especially comes into this candidates, whether the previous section also, the presentations they were talking about. Independent type of candidates has its own characteristics to design the DSP. We wanted to be more precise towards manufacturing and equipments where the bioreactors and then fermenters and collaborate with the DSPs. So the scale up becomes much more efficient with our clients. If I say like I can do even a bioreactor, DSP, and then a downstream process complete system, then may or may not be completely efficient. We are not at the stage, but the integration is we discuss with our partners in a DSP partners, and then a design from the beginning. That's one of the unique process that we provide is the digital twin technologies. A digital twin technology provides an opportunity to the scale up the construction where you can the save time and also the project planning when you do the digital twin technology. So series of from the beginning to the end, how it starts, how it ends, we can design. That is with our collaborators. So yeah, I also like to show you some images and like our the projects that what we have done recently, just to some of the facilities that what we have done is like over 4 acres, 5 acres, like a massive facilities that what we have done. This is one of the project that what we have done is like it's a chocolate manufacturing facility, GMP facility, globally known facility. So complete design, development, integration, that's what we have done. That's one of them. And the ethanol project, it's a different scale that what we have. It still is a pilot scale that is going around over total volume of over 500,000 liters capacities. That what we have done. What we are doing are ongoing projects. This one. So yeah, the designing of the ethanol productions is non-biological, that comes in a different scale. Yeah, that's about we provide complete end-to-end solution. Again, as mentioned, we align with our partners.
Great.
So, of the equipment.
Could you talk briefly about the how you work, how the collaborator can work with you. You mentioned about the first step will be the NDA, and can you go through the general process?
Yes, definitely. Like a NDA, we have confidentially protected, and then also we work on the cost efficiency and also lead time. If somebody is capable enough to provide that aligning to us, definitely we are happy to discuss. And also, like, it's not just a cost efficient for our company, because it's, again, when the collaborations comes as a bi-directional, like a kind of an we need to earn some kind of a provision as well. So it's not only for us, but it's end user. That's where we keep it in mind. By collaborating, does it really impact on the end user? Because if you see the progress in the bioprocessing bioeconomy is increased over 2022, is 2020 increasing further. So most of the company, when we discuss with financial constraints, is extremely difficult. And then how a company going and talking to them, we are providing a solution, but it costs you like a massive amount of the upfront capex. So that's where we work with how the OPEX and then your ROI can impact on it, so that they can sustain for the long time. That's where we are coming up in a AI technology where we can decrease the number of employees. I'm not meaning to say that we can cut down everyone, but the efficiency enhancement we wanted to see. That's where the vision integrations, that what we are bringing. The workforce management, just an example, that's our own company. That's what we have. Different verticals of the tech family. We have already provided the technology for different companies right now, because pressure vessels monitoring is always a kind of a challenge. And then in a tall, like the length, how you can go tall every time. Can you climb up and go down in the process? No. The piping and pressure management, and then also process management, it requires a huge support, whether it's an internal vessel management or outside vessel management or facility management. That's where I'm talking in relation to the scale up as well. So that's where we have our AI technology. The cameras are specially designed for this type of technology, can withstand 1000 degrees Celsius, which we have used in space purposes, and also minus 200 degrees Celsius. The technology can be worked. So it can go inside the reactors, whether it's in a temperature sensitive or your turbidity or viscosity or your different types of internal environments. And then our IoT, I think previously somebody was mentioning type of IoTs and sensors is valuable. So we are working on upcoming the like a bioprocess engineering is also includes, which is not existed in a large scale in a previously quantitative measurements. So we are working on a little bit of innovation towards sensors as well, developing different types of sensor predictions.
Great, thank you. Let me introduce our next speaker, John Ellersick from Next Rung Technology. John will talk about accelerating the commercialization of sustainable technologies. John, are you ready to share your screen?
Thank you, Ning. Thank you for having me and having us. My name is John Ellersick. I'm the president of Next Rung Technology. As Ning said, we focus on accelerating the commercialization of sustainable technologies. And we thought we would give some introduction to ourselves and the things we do, but we would do it following some of the themes we saw in 2024 and what we see emerging for 2025 to add some sort of content and the type of topics we talk about and guide folks through. So this is really what we're thinking about, is how Next Rung Technology, we work with our partners and our clients to navigate these times. So the quick introduction to Next Rung Technology, this is sort of my favorite picture and slide of us. We're an engineering consulting group, now around 20, almost 25 engineers and analysts. And we work on commercializing sustainable process technologies. This is 5 of us standing on top of a facility that I had helped develop with Justin in sort of the first wave of bioindustrial technology, making succinic acid with Miriant. And we were nicely brought back in when that was acquired by subsequent companies to help commission and start it up. And I like to show this picture because really the idea of scale up that people talk about and the idea of what a techno-economic model is trying to do is to know what your process looks like at scale and to really know that's where you're going and to work back from there. So this is what 400-meter cube fermenters look like, industrial production of bioplastics and crystalline products. And again, to also emphasize that we are interested in this, we do this because we are interested in sustainable technologies. So we were started in 2017. We've had over 150 customers now over the last 7 years. And we do the set of services and activities that folks commercializing emerging technologies need to do. And that's different for different people. But it's basically this whole set of activities with a lot of folks. We're helping them do planning and road mapping. We have some very good R&D folks who know how to translate that small scale benchtop piece of equipment, or even a sous vide or a hot pot that you're using to do some form of reaction into what that means industrially. The core of what we do is a lot of project development, a lot of FEL 1, 2, 3 scoping activities, project delivery, including for smaller projects up to say 20, 25, $30 million, we can fully help deliver it. And when projects get larger, we help folks get their projects and their technologies in shape, still commonly through the FEL process and risk assessment programs. And then we often function as an owner's project manager, owner's engineer, help folks do the outreach to find engineering companies and the right partners for their larger capital projects and stay with them the whole way in delivering it. And I guess I'd say the last thing we're really well known for is in the sort of consulting modeling space, we do a lot of techno-economic analyses. We do them in a bit of a different way that's very visual. It's thinking about organizational goals and teams. And it's being done by folks who have built, scaled, and operated technologies, which I think is different from, sort of the individual providers with big spreadsheets. We work across sustainable process technologies, but we're here today because about 60 to 70% of what we really do is industrial biotech, next generation proteins, foods, cultivated meat. It's a very core part of our business, and it's important and impactful from a sustainable technology perspective as we move beyond fossil fuel as a feedstock for a lot of these materials, as we think about onshoring American manufacturing of materials that are produced elsewhere, and as we think of more sustainable means of agriculture. So that's us. In brief, we're located at Greentown Labs in Somerville, which is an innovation ecosystem. But we're throughout the country and a lot of other places, and internationally, we work in Canadian ecosystems in Japan and Korea. And for instance, at New Labs in Brooklyn, I'll be down there over the next 2 days for the Hack Summit, if any folks are going. There's a big biomanufacturing piece. I'll briefly flash, this is not a complete glimpse of all the sort of processes and products we've done, but this is sort of the generations and waves of industrial bioprocessing from biofuels and chemicals, moving on to a lot of replacement proteins and foods, textiles, materials, plant cellular ag, animal cellular ag, as well as having worked on a whole sort of the gamut of fermentation platforms and technologies, as well as emerging technologies in areas such as low-cost feedstocks, scale-up facilities. So that's a little bit of an introduction to us. And then I would get into a little bit our themes from 2024. So in 2024, we pretty much every quarter go out and talk to folks, talk at conferences and try and share our thoughts. The first thing we did in March 2024, I should say, and what we will do again in March of 2024, is talk a lot about ideas about when to go to contract manufacturing or third-party manufacturing, when to think about building it yourself or finding other partner routes. We called that presentation CMO No or CMO Go. And you can find that on a website. We did it. BioPTP hosted us. BioPTP has a nice open CMO database. And we collaborated with them to do this presentation. So the presentation is thinking about what it looks to scale up. And you'll see as we talk about scale up, we'll sort of view the whole process in a way most folks on this call will find familiar, which is a series of moving from one level of technology readiness to the next in biological processes that involves organisms, then fermentation and downstream processes and customers. And it goes stepwise from lab and bench to pilot to demo to commercial in a simple description of what you do. So your path to commercialization, surely, and the roadmap that you're following is hugely impacted by your starting point, whether it's early or mid-stage where you are. And you start to run into a bunch of questions as you're scaling and commercializing. First one many folks run into, and I encountered this maybe 3 or 4 times myself when I was working for technology startups, is whether to build a pilot plant or use third-party resources. And this is that first encounter with the idea that if you are venture funded or have other investors, they may not be interested in putting money into steel. However, if you go to third parties at the end of the day and don't develop your own facilities, you may be left with nothing or left with costs that extend if your time extends in piloting. So that's the type of first decision folks run into. A second one of a greater scale, and this is really where CMOs are often considered, is around demonstration scale, meaningful production of materials for the market, meaningful production of materials for sampling, or actually long-term production to serve the market for 1, 2, or more years. So we call that's the CMO Go or CMO No-Go point. It's really a make versus buy evaluation. And we have a strategy where we think a lot about what a grassroots facility would look like, overlay that design onto your CMO options, and use that to really conduct a sort of a make versus buy evaluation, along with pros and cons. So in March of this year, as we looked at the evolving CMO landscape, we identified some of the trends that were coming through. So one of the great trends that's going on is what was once invisible and very hard to find is becoming much more clearly defined and accessible. BioPTP created a nice open database. Capacitor created a nice open database. These resources are now available to find. A business that once sort of lived in people's black books and notebooks is now much more out in the open. And we think that goes a long way towards debottlenecking the scale-up landscape. As I think most folks here would know, the government investments at the state and federal level are catalyzing sites to fill in missing sizes and scales. For instance, the BioMade network or other states are also investing in sites for scale-up. At the same time, there's a lot of sites who have CMO capacity. They would like to monetize better. They would like to fill. It might be surprising how much underutilized capacity is out there at the smaller and larger scales. And that's the idea of these open network linking tools, because it's as much a matter of connecting folks as it is building more capacity in many ways. We do see things, the regulatory hurdles still exist, but people are continuously moving through GRAS, USDA, FDA approvals, which is very positive. And in 2025, we'll be doing some thinking and putting some content out there. As much as there's a stated shortage of assets, I think there's really a stated shortage of rationalization of assets. And if you were one person or company who had all the assets that even exist in the country today and rationalized their use, I think you would use them very differently than we are. As driven by funding, money, and the imperfect way that folks with technologies have to encounter CMOs. I won't spend that much time further on this slide or topic, but when we help people go to contract manufacturing, We really think the first step is sort of charting, clearing up goals, a proper outreach process. And there's some talk about this yesterday. This is essentially a version of a tech transfer process. And this happens if you're working with engineering companies or if you're working with CMOs. At the end of the day, this tech transfer process has to happen. So we really encourage people to do it at the start of the day and use that as their bidding and outreach documents to give them a set of multiple options that have seen a professional outreach and open up commercial options for them, as opposed to heading towards one or two options without the same information prepared and filling it in at the end of the day. Deciding where and how to go is rarely just simply economics. There's a lot of other contractual things we keep in mind. But this is one of the first topics we talked about in 2025. We're doing another session on it, a workshop in in 2024. We're doing another session in March of 2025. We'll have a link for folks to sign up. And if you're on our newsletter, we'll tell you when that's happening. So the theme around CMO was there's still a shortage of capacity, but it's easing due to the publicly available information. Clients are finding sites, and I just want to include a couple pictures of us. You know, one of the things we'll highlight that differentiate us is we're not just filling one space or another in the ecosystem. We're a very hands-on oriented set of engineers. And what we love to do is go on site. So this is us commissioning and starting up some facilities. And not only do we help folks find the right CMO and the right way to address them, but we'll go there and go with you and help you attend to these important runs if that's helpful to folks as well. So that was our first theme. In the second quarter, important theme I think most people know evolved, which is the impact of government support in our sector. So the DIBC program was announced. They finally, in the spring, people were submitting white papers to get funding for development of projects and ultimately potentially money to build them. And over the summer and fall, the Department of Defense announced 34 awards. That's the Defense Industrial Biomanufacturing Program. Additionally, we're seeing build out. So Biomade has announced their expansion of the pilot plant network, which is very exciting in multiple states, filling some of the gaps, providing an impetus and acceleration for the bio industry. And this funding is impactful and important because most of the folks here know on this call, investment in the space peaked in 2020, 2021, and has been dropping off literally for almost 2 to 3 full years of quarters. So government funding, government opportunities have stepped in meaningfully where private funding has slowed down. And they've slowed down as due to the interest rate, as well as having perhaps made a lot of investments in the sector as well. So those are the second set of trends we saw in 2024. Trying to move this sucker here. And just for us to state, like some of the things we were fortunate to do here include working on the Schmidt Futures program, some Biomade funded projects, including this is us developing very deeply integrated TEA along with a life cycle analysis that really gets to the answer of how to balance capital investing, environmental sustainability, and really gets to ideas that support green investing. Furthermore, we did some nice work with the Biomap Consortium and some of the DIBC related projects. So that was our theme for 2024, number 2. The third thing we saw dropping in the fall on the cultivated meat industry. This is an area where we've done a meaningful number of work with small, medium, and large players. And we were presenting in Boston at the Industrializing Cultivated Meat Summit. And we're talking a lot about healthy scaling or scaling healthy. So this is our way of thinking about that these technical tools, all the tools for scaling, for designing plants, for planning your progress are not only technical, they're organizational. We have a database of somewhere between, let's say, 50 to 100 industry projects that we pulled together to evaluate economics, costs, and timeframes across that realm of scales. And we looked at this and presented this information to think about what's it take in time and money to scale these technologies, scale emerging technologies. This data set right here is that same time frame for the top 10%. And you can understand and think that it's it's feasible to do some existing lab development in 15 months to move to pilot scales. These are the top 10th percentile timing in months. And if you stack them from start to finish, you start to see if each of these is a 5-year block of time. It's really a 7 or 8 year time frame in the best case to bring bio products and bioprocesses out into commercialization. And that sort of corresponds with the investing window of venture capital and explains sort of why it's a challenging form of financing for bioprocesses. And now these are successful programs. This happens to be what the 90th percentile in duration looks like. These are, again, from clients and processes and projects we know, the 90th percentile duration of successful programs. So for instance, we have folks who've gone out and produced product at the CMOs for 5 years. And that sort of turned out to be the right decisions, for these types of clients that we help guide them to. This presentation was prepared in particular for cultivated meat and all proteins audience. And the message there was the trajectory here is actually tracking along the time horizons of some of the biochemical commercialization. If you think about the first wave of industrial bio happening that 2005 to 2010 timeframe, we're seeing people build plants 2 and plants 3 today. So we're really, preaching to folks that this is a good time, especially in those longer adoption technologies, to practice good capital discipline. We have a set of technical tools we bring people through, for example, road mapping to determine how all the pieces of commercialization fit together and what sizes and scales make sense for your plants. We help take people through the FEL process and understand the purpose of those stages and why, and do proper stage gating. And then, of course, people know do a lot of techno-economic modeling and analysis. And that's really all not only technical, but organizational to help technical teams and business teams get on the same page and chart a path towards commercialization. So our message and theme from Q3 2024 was, continued capital discipline. I've built many of world's largest plants of one type or another, but really quite often it's smart to, build things as big as they need to be to prove the things you need to prove. And so we said, instead of measuring twice at this point in time, you may need to measure 3 times. And then our final theme for 2027, and I'd love to make this bar move somewhere, but it won't, is the ecosystem. So we're embedded, as I mentioned, in technology ecosystems. These are the ecosystems that create exchange of ideas and real progress industry in our experience. And so a couple of things we did, we gave techno-economic analysis bootcamp for clean tech startups and director capture. And I just like this picture. You can see this is actually a slide photo of us giving the presentation, and sort of one of the things we like to highlight in our TEA presentations that we are people who build and run plants. We're experienced in operating them. We have real capital cost information that's up to date. So you're getting the perspective that's realistic and up to date, and has supported a lot of capital investment in companies based on just the realities reflected in our TEA models. We nicely presented Harvard Business School Climate Summit right after Halloween. So you can see our Halloween candy is on the table. We're always thinking about scaling important technologies from future food tech. We do a lot of road mapping with clients. Here we are doing this with the Korea desk. at the Cambridge Innovation Center. We've also done this with the Japan desk. And we do a lot of content to put it out there in the ecosystem. If anybody's interested, we're happy to. We'll host these throughout the year, or can take you through some of these programs on risk management. Come to the US market. You saw the CMO go or no go, and our road mapping session. So our fourth theme from 2024 was supporting the ecosystem. It really takes a village of people collaborating to make a startup scale, and a good, strong ecosystem is essential for any individual company and for our industry as a whole. So that's our sort of who we are, our four for 2024. And then we'll give you a quick take on 5 themes for 2025. I guess we call this the bio baton because we're passing the same theme from 2024 to 2025. There continues to be strong federal support for biomanufacturing. Folks are out there trying to understand and think about the federal government's priorities with a changed administration. But many of them have always aligned with biomanufacturing. It's always been a well-supported set of programs in both blue states and purple and red states. It's really a purple issue. It's good for American agriculture. It's good for our technology leadership. There's a very strong, very clear interest and message being sent by the incoming administration about onshoring of manufacturing. And it creates great jobs up and down this country. So we believe that support is going to continue. That'll be one of the major trends of 2025. The corollary of that, as sort of in an advising way, is avoiding grant chasing. There are a lot of opportunities out there. We really work with our clients to make sure they focus on the right steps. They build the right size of projects. As I noted, they're working with the right set of partners. Having that roadmap of where you're going, understanding the economics and the economics of scale are critical for the decision-making about what projects to do next. And I will always highlight this good question is, would you do this project or activity if there was no federal funding or grant? That's a great question to ask because sometimes chasing grants can pull you off of where you need to get to. You've already heard and gotten a sense that we really think, robust project development, starting with chartering and good stage-gated execution, is a little bit slower than really fast execution, but it's critical at times where capital is hard to come by. We love risk management and risk assessment as a tool, not just something to avoid, but it's a very good organizational tool to bring a set of folks through the activities of scaling. And we just suggest, we have many folks who come to us who have perhaps aimed and fired and gotten sites even before they figured out what their projects are, but the steps remain well considered, and we suggest following sort of the processes that exist. 2 more trends that we see being left out there, and I'll take a little time for questions. An execution squeeze. If you're trying to design, engineer, or deliver projects right now, I think you'll notice it's hard to find real resources. Folks are really very busy. There's a lot of larger projects and federal projects going on right now, and we predict that is going to follow through the completion of 2025 into 2026 and move from the design and engineering through execution and supply chain. So that's a predicted trend for 2025. Supply chain pipelines are filling. And what we'd say is just advice there is that doesn't mean you move beyond stage-gated project execution, but understand your fully integrated projects, where the long lead times are, and make sure you're working on the right things. Excitingly, fermentation capacity is coming online 2025 and 2026. We expect additional capacity online and the shortages to start to ease. So those are our thoughts. Those are our 5 for 2025. We'd love to hear what you think about it. Many of these don't really change the idea that of how you would approach things. And again, for CMOs, for instance, to use competitive bidding as a greatest tool for options and negotiation. And we're excited about the next year. I can tell you two things we have coming up over the course of the year, and if you're registered for our newsletter or if you contact us at info@nextrun.technology, we'll keep you in the loop. We'll be doing a refresher in March of the CMO Go or No-Go content and as a workshop, and that includes reviewing any CMO outreach documents, providing people with a template, and giving you feedback on your approach. April is our audit month. So in April, we're going to offer TEA audits. Again, the same thing, a 2 or 3 hour sort of review of your audit, critique of what's in your TEA, your capital cost database, and give you feedback in April. And I will stop presenting here. We're Next Rung Technology. We are committed, really, and have the goal of helping us achieve a healthy and sustainable planet. And if you stay connected with that QR code, you can get our newsletter.
Thank you, John, for a great summary of the commercialization process. Have one comments in the chat. John, could you post the link to your CMO Go No-Go presentation?
Sure, absolutely. I will find it and put it in the chat. It's a BioPTP webinar.
Any other questions from our audience? John, I have a question.
Yep.
Seems like you have worked with a lot of different types of projects, different products. What do you see the main challenge when you help them to scale up their technology from bench all the way to commercial?
So I think it's really critical to understand the maturity of your technology at each step of your process. And we sort of handle this as a risk assessment, but it's a knowing where you are, understanding if you really truly have representative data, if you've been doing representative processes. And as you're looking to scale, we sort of have a tool that helps quantify. It comes from due diligence that we've done for VC investors. How much are you scaling at each step? Have you operated in that manner? Or is there more technical work to do? And you're going to be designing a facility or plant that's going to be operating in 16 to 24 months from now. So your technology will be in a different place as well. So we sort of talk about understanding technology status and that it's moving and designing facilities for day one and year two. And that's really how you design a facility that achieves your goal when you get to your business case, while it supports your technical realities where you may be today or tomorrow.
Thank you again, John, for presenting here. Now let's move to our next speaker. Adrienne McKee from Hawkwood. Adrienne will talk about real world expertise, accelerating biomanufacturing. Please take it away, Adrienne.
Thank you very much, Ning, and thank you very much to Deepti and the other hosts, Leah. I really appreciate you guys for putting this on and organizing everybody. It's my pleasure to be here today, and it's a tough thing to follow John and Next Rung, but I'll try to do my best. Let me bring up my slides, and while I do that, let's see, here we are. I just wanted to say another thank you again. I've been in this space at a number of startups. I got my start effectively in industrial biotech, working out of Jay Kiesling's lab back in 2008, right as synthetic biology was really becoming a thing. And I had the great opportunity to move over to a startup as the day that it IPO'd at Solazyme. So I've been in this field for a minute now. Really appreciate some of the insights I've gotten from folks along the way, and that's part of what Hawkwood is here to do. So is to help aggregate those insights and share them, and also connect the right people at the right times. Let me make sure I'm in present mode right now.
Yes.
Okay, great, fantastic. I'll jump in here. So I think this is a really it's nice to start off a talk with getting folks to agree on something, and I feel like this is a pretty simple one meatball for us to throw out here, that scaling industrial biotechnology is challenging. And having worked at a number of startups, I can say this for sure, that startups often lack the expertise, but also the capital to access it. And so as you go through these phases of development, scale up, and growth, there are different parts of an organization that needs to exist, and there are different types of people that need to be in the right place at the right time. Oh, sorry. I'm just getting used to the bots here. Okay. So Hawkwood is a consultancy that's dedicated to biomanufacturing writ large. So within the bio landscape, collectively overall, we're really focused on those areas of biomanufacturing that are focused on fermentation. We are starting to grow that a bit, but even within that part of the technology, we cover industrials, food, agriculture, and energy. One of the values that we bring, or the advantages that we bring, is this diverse constellation of specialized skills that companies need as they're making that transition from development into growth. This part of the valuation curve is really where all of the work goes on, I'd say. And there is quite a bit of it. It's beyond at the point where you have sort of an initial proof of concept, and maybe even some very good strains, a very good market idea, et cetera. There's a lot of expertise that needs to come into play. And one of the things that Hawkwood has been able to do is assemble a very amazing group of experts that are very good at their particular part of this pie. And so in some of the use cases that I'll get into, case studies, show how we've been able to lean on this network that we have, that we can basically pull in the right person at the right time. We have several different areas of things that we help companies do, and they fall largely into three different buckets. So I'll start with strategy and commercialization. And within that, it's everything from looking at the market, how big will this be, under what circumstances, what kind of partnership should you go after. We also get asked quite a bit to do some internal industry connections for voice of customer interviews, to go find those people that have knowledge but are maybe not putting it out on a shingle somewhere easy to find. We work a lot with trying to establish pricing for early companies, as well as have folks on our roster that can inform on regulatory as well as IP. And in the engineering and manufacturing side of the firm, we also do techno-economic analyses. Our offering is really sorted at the sort of FEL 1 stage, so pretty sort of early on in the process, but very fundamental for getting a groundwork document that is critical to be able to share with early investors, et cetera, and then also being able to be used as an ongoing living piece of IP that informs the rest of the company. We'll get into, I'll have some use cases here in a second on both scale up and product and process development. But within the network of folks that we have, we are able to deploy engineers who have experience with working at different CMOs, but also developing bespoke processes for a host of different application areas. And then perhaps maybe not germane exactly to this call right here, but just as an FYI, we do a bit on capital investment and acquisition diligence. Some clients will come to us asking for specific information on either a space or a particular company. And so who are we? Well, this is me. As I mentioned, I was doing synthetic biology before it was cool. My two partners are Tony Day and Richard Kenny. They founded the firm in 2017, 2018, really at sort of the as Solzyme was wrapping up. At the time, the firm was really just the two of them, but we've grown now. We're actually 10, and the slide hasn't been updated as we have a whole nother member that we have yet to add. But we, in large, the full-time folks on the team are operators with broad experience in scale-up and commercialization. We have a host of engineers, sort of more junior engineers, and then in-house, and then a number of engineers on our team within the council. And that leads me to describe the council itself. As I mentioned, we're able to window in areas of excellence and people who have deep experience in the space. We have a connection with, I'd say, 35. This is actually on any given day closer to 40 council members who bring loads of experience from CP Kelco or Kelco in all of its various forms from DuPont, Merck, Tate & Lyle, Unilever, etc. And we work on projects both with the folks inside the office and this cadre of the luminaries in the field in a way to be able to put together a bespoke crew to solve the problems that we're being asked to address. I think this is pretty relevant for the cast here. The manufacturing scales is basically everything. Commercial scale can be much larger, but we feel very confident advising, even from the lab all the way up to, hundreds of thousands of liters. Processes cover all types of fermentation, including gas, and then downstream as far as how long is a piece of string. Folks in our space, in our network, have worked on products and in applications in effectively every market area that industrial biotech touches upon. And so if it's something that is not pharma, it's pretty much something that we're able to jump into. So one of the things I wanted to do in this discussion was present a few or I'd say case studies of things that Hawkwood has done on behalf of clients, and sometimes rolling up sleeves very, very much side by side. The first one I'll get into is a small case study for a project that we did adding extra engineering team members to an early stage startup, and I'll get into some of the benefits of that. We've also acted over the years as what I'm calling a Swiss Army knife support for a fermentation startup, and in a wide variety of different elements, and I'll get into that in a second. And then third, just an additional case study illustrating pulling together different expertise. And in this case, it was working on a techno-economic review that was in service of both a major multinational and a startup, and being a bit of a third party between the two of them to come together on a solution going forward. One of the things that I'm doing here with the photo is having a nod to some of the applications that were going on. It's a bit disconcerting. We weren't actually making computer board chips, but we were helping a company develop a process to develop a monomer that would be used potentially in a conformal coating, for instance. So in this case, we worked with an early stage startup, providing additional layers of expertise on their DSP. What the team sought was was going from effectively a really small amount of product and understanding that they needed to be able to develop a scalable downstream process. That needed to work hand in hand with the techno-economic model that was also being supported by Hawkwood. So what we did was jump in and deploy a fractional development team. That was one person visiting their labs pretty much on a routine basis for a number of months, as well as a senior engineer acting as sort of orchestrator from the sidelines. So we participated as owner's engineer here. We built an in-house model for them, and then we facilitated engagements with air EPC firms and also JDA partners. One of the things that led to was both a scalable process for one, but they were able to produce the samples that they needed for validation with customers, and then that also helped them with conversations that they were having with their investors. Overall, one of the value propositions that we bring to the table is that we can save companies time and cost in sort of getting to a process, but then also in figuring out who to have in the right place to be able to do these things. I've worked at a few companies where, and I think we might not be in the same sort of economic times as maybe 10 years ago, but it's hard to hire and it's hard to keep folks around, and to be able to window in people, get a job done is certainly a value. Next up, I wanted to sort of talk about this work that we did with a startup, taking all the way from very, very early stage. We started working with this organization when it was, I think, 1 or 2 people at the company. They had an idea for a product, and Hawkwood was able to jump in and act as a Swiss Army knife. This partner, we worked with them pretty much all throughout their seed and Series A, and a bit now into getting to the B stage. We were able to fill some fractional roles across a number of different areas, including science, engineering, business development, but also something that doesn't necessarily raise to the top of people's minds is also finance. They also were looking for fundraising support. How do you go talk to investors? What do you need to say? What do you need not to say, etc. And over the course of several years, we worked on narrowing what the product market was going to be, who needed to be there, in how to sort of stage those hirings. Again, as I mentioned, fundraising support. Techno-economic modeling is never done, but we initiated the work plans there and have been started that and oversaw it for a number of years. IP strategy, put that forward and drafted their initial patents. And I mentioned now, I believe CFO services are something that we are actively doing for a number of other companies in addition to this one in particular. We're really excited to see this particular client go forward in the world. Our involvement is one of the things that I think is tough when we sort of window in, is that we also, under the greatest circumstances, when everything is going as we'd like, or as our clients would like, we also window out. And so we have made the transition to hand over these jobs to more full-time people in the actual company, but we feel very good about how we've been able to set them up, hopefully for success in what is making a really amazing product and some pretty happy clients. And then lastly, I wanted to talk about a paradigm in which we helped out both a multinational company, in this case, somebody who's really interested in paints and coatings. And they were developing a joint development agreement with a much smaller company that we had also been working with. So collectively, the partners were looking for a way forward and come to a point that had a few questions in the road. The larger company was really much interested in assuring that the techno-economic model was sort of buttoned up, but also very much interested in where in the world would be the best place to build a facility. And those kind of situations are the answers to those are really material, because that larger organization was going to need to make some pretty big asks of its management team to put forward more money. So as anyone in a joint development program knows, just getting the first one is really part of the journey. It's moving that forward and taking those products to market. That needs a lot of buy-in on the larger corporate side. And what we were able to do was provide a package that validated the smaller partners' economics and walked the larger company through a set of scenarios about how they should think about situating a sort of on-ramp for development of this new product, and then thinking about where in the world would be material to build this facility. In this, I'm getting a bit into the weeds, but we looked at plant cost scenarios and location recommendations that got pretty detailed into utility costs and I'd say labor, and also just various other things that are really important to consider when either working inside the US or in various different countries on a country-by-country basis. Happily enough, this development program progressed to the next stage, and they were able to pick a sited facility area where they are thinking about building this facility, and that sort of question was resolved. So these three vignettes, so to speak, provide a bit of flavor of the kinds of activities that we do, that we're really happy to do. And it sort of thrills us to be able to make industrial biotech work. I have a few more minutes here. I wanted to talk about a project that I actually worked on pretty in depth over the course of 2023. And one of those areas was talking to the different council members that we have on our team, but then also reaching out to a whole host of other company, friends of friends, etc., talking to them about their scale-up experience. I've seen a lot of media sort of calling out, the difficulties in scaling, particularly, the reproducibility of going from, say, 5 liters to 100,000 liters, etc. And there's sort of, it seems like seems like a lifetime ago that this was actually a debate of saying, these sort of things don't scale. And what we really were finding as we were having these conversations with the engineers on the team was that really wasn't the case. Pretty much everybody that we were speaking with said, hey, once we were able to get to like a reproducible 5 liter, getting the process to work at much larger scales was actually really not as problematic as everybody was talking about in the space. So we wanted to say, well, okay, well then why is this so hard? What else about this was difficult? Why is it so hard to be both a startup and doing scale up? We talked to a number of folks about their technical goals, the resources involved, what was the outcome, and then sort of what happened in both a good way and what didn't. And one of the, just as a note, drawing 2 sets of arrows, is that we talked to a number of people about moving down in sort of scale down models as well as scale up models. Overall, I think we ended up connecting with about 20 or so experts. There were CEOs, founders, senior and junior R&D leads, and this represented about 32 companies overall. At a high level, the flags here, I think, are actually applicable not to just industrial biotechnology, but because that's the space that we're in. I think it's pretty important. And also, I wanted to say that pretty much most of the survey that we did and the conversation were in groups that were focused, that were largely in sort of the VC-backed space. I'd say by and large, one of the largest challenges was aggregating technical debt, basically running faster than the technology was able to move. But we also found a fair amount of misjudging product and market fit. And I'm going to say that CMO tourism, particularly in like the 2021s and 2022s, was a real drag on companies. Finally, and this is maybe a bit of a throwaway, but very material to us is lacking the right stuff, just not having access to either the right equipment or the right people or know-how. One of the reasons that we did this sort of effort was for us to figure out how we could sharpen our offering to the space and also figure out, really what's needed and why. So what we did is put together from this a checklist for startups for things to think about as you're going, from that development stage to that transition. Maybe you've gone through the ABPDU and you're starting to you're more than starting to think about what is next. And so happy to share these. I don't need to talk to them in depth here, but very, very excited to have an offline conversation with folks. Very much as similar to the bullet points that I highlighted earlier, the major areas that we are considering as super important are in that first and foremost in that product demand and market fit space. In the manufacturing and economics, particularly looking farther down the road to make sure that there is margin on the product that you're going to be developing in several years out. Continuous DSP, you're going to be able to show that. Resource planning writ large, and a bunch of sub-bullets in there. And with it in respect to the R&D stage, a number of different things that sort of are on a case-by-case basis. So I think, alas, this is definitely no guarantee for success. But as a number of us have spent many, many, many years, and collectively the council has spent dozens, if not hundreds of years working with startups, we think that this is a good start to consider. With that, I will jump to our contacts page and just pause here and say thank you again to Deepti and the organizing team for putting this together.
Thank you, Adrienne, for the great overview of what Hawkwood do. I don't see question yet. They may come later in the chat. So a question from myself. So you work a lot with startups companies. Generally they don't have their own facility.
Mm-hmm.
Do you see any difficulty for your customers to find a scale-up facility to, validate their process?
I think it's, that's a really good question. What we have seen a shift in the space. I'd say that the answer a couple of years ago was, yes, it is more challenging. And I think that there is additional capacity that has shaken loose for a whole host of reasons. A number of different organizations have surfaced that there are other sort of manufacturing facilities available. Folks across the country and then in different parts of the world have raised their hands to say, actually, we do have some capacity. So that I wouldn't say it's necessarily solved because all of these things are not plug and play, and everybody's process is going to be a bit specific. Where we get really more into the weeds and into a bit more of a mess is in the downstream, and sort of having that equipment be available, and so companies don't need to fly or create their stuff from one country to the next I think that one of the things that we are really monitoring closely is how this additional capacity from various different players across right now, India, not necessarily just India, but sort of other parts of Asia, etc., what that's going to do to the costs of runs, the availability of runs, and things like that. So we monitor that kind of thing and have a pretty good running internal pulse on. Wouldn't say completely comprehensive by any means, but are able to inform on those kinds of questions.
Thank you. So you mentioned, so you're coming from Kissling's lab. I'm wondering, do you also work with federal funded projects?
I'd say yes, we do. But it is directly via our clients that are doing that. We have a number of folks that we're helping on various different DIBC awards, and we're really excited by that. We are also working with some other parts of the, I would say, the ecosystem that are funded through either NSF or things like that. Hawkwood itself is not a direct grantee yet. I think probably we have a bit more lobbying to do to be able to reach that status. But we are active in a number of activities right now and hope to grow that part of our firm.
Great. Good to know. All right. Okay, I don't see any more questions. Let's thank our speaker again. Thank you, Adrienne.
Okay, thank you very much.
Okay, I think there are only a few more minutes to move to our next speaker. All right, I think I can go ahead and introduce our next speaker, Sergio López from BIOANG. And Sergio will talk about accelerating bioeconomy adoption through AI-driven feasibility analysis.
Yes, thank you. Thank you very much. Thank you for this meeting. It's really an honor be here talking about this topic, especially with wonderful specialized like John, the other engineerings. Well, we are a startup, so we are going to talk a little about who we are and why we are doing this. Okay, we are BIOANG, and we create this startup with the vision to help companies in the agro and food sector to adopt the bioeconomy technologies to get value and transform the organic waste into bio-based products. So a little about us. We are a team who has more than some of our members has more than 20 years helping agriculture companies in business in the US and Mexico. The other teams, we have more than 10 years working on bioeconomy and bioenergy projects, some research and development, some technology adoption, industry level, principal in Mexico and in Spain. Some example projects is wood waste to energy, wood waste to bioasphalt, hydrogen, pineapple waste to textile fiber for research and development, etc. For me, like 7 years ago, I founded a startup, a research and development startup in an MIT enterprise program to develop a biorefinery to process mango waste, and I got a patent to extract some polyphenols and pectin from mango peels. And we are working in some masterclass. We had been part of the experts help the Mexican government to develop the bioeconomic strategy. And for this project in specific, for this thesis, we are collaborating with FermenteQ lab experts on techno-economical analysis. So we have been helping some companies to adopt technologies to transform their organic waste into bio-based products for many years. And we realized that we have some challenge, and at the same time we have some opportunities. We know that there is a lot of biomass potential that is unused now. It's in a lot of food chain production. But as we see, there is a challenge on the technologies and the adoption. We found two principal challenges. One is like the technologies are very complex. It's necessary to be really specialized on one technology. But at the same time, the biomass it's really heterogeneous, and in all the cases present really unique variabilities. So it's hard to see to find what technology could be the best for any cases. And if you are an agro company or food company and you want to go for a specialist, normally you will go for a specialist on biogas or a specialist on biochar, fermentation, etc. But it's like if we see the picture like in the medicine, we have the general doctor, and then the general doctor conduct to specialize to make a solution. In this case, we found that we only have specialized, but we don't have too much people who can focus on help companies to advise on what could be the best specialized for the cases. So that's the reason that we develop some services and we develop a methodology to find the best matches with biomass and technology, considering some techno-economical variability, social variabilities, the needs of the company in specific, etc. And we offer some service to detect what could be the best opportunity with the biomass waste, some feasibility studies, research and development projects. That's our first or main services. And one important thing is like we are not gods, we are not doctors or in all the technologies. What we do is we create working on these years, we create a network with specialized with lab specialized, company specialized, to let us make a platform to once we detect one opportunity with some specific technology, we work with some specialists on that technology. So we developed this methodology, but we realized that we have some challenge, and these methodologies John, for example, talk about the challenge on the scale technologies. There is a big challenge in this case that it's necessary make a lot of techno-economical analysis, and this techno-economical analysis tends to be expensive and time intensive. So it's not real accessible for some companies. It's normally this kind of service tends to be accessible only for big companies or companies, but making that makes a little hard to massify, accelerate this adoption. So that's why we are applying AI integration on our methodology to try to make it massify and make it cheaper and with less time to get access to more companies they can they can see and they can take advantage of this bioeconomy technology. So we realized that using AI in some process, some specialist techno-economical process, we can reduce at least 50% cost and 50% time. So using some databases that we collect on these years, so that's why we are focusing now on use AI, these kind of technologies, and we want to make several products with AI focusing on some feedstock, especially for example agricultural residues, nutshells, etc. Some forestry waste, and in food processing waste. We realized that we can make some specific products using AI to make easier for the companies, for the agro companies and food companies, find the potential that they have with the biomass waste. So in the meanwhile, we realized that it has a huge impact on the community, because if we get to achieve the accelerate the adoption of these technologies, we could create a significant impact on the climate change. We can create more employees, and we can promote more value and local chains. So our next steps is we are creating our first MVPs or first pilots with using AI. Now some experts from Berkeley Lab, we are working to develop our first pilot specialists in choose the technology to produce heat and energy, focusing on 5 of the most common technology like pyrolysis, gasification, boiler, and we are preparing a project to present in the DOE topics for SBR to make our first pilot. So we have some testimonial cases that we can help, or why this we are focusing create tools to help companies to choose technologies. Like some companies, for example, in Mexico, we achieved to transform to transit use oil to get heat, to change it for biomass and save millions of dollars in a short times. Millions of Mexican pesos. Or for example, these cases we work with Deutschland company called Net Energy, specialized on gasification. So we achieve to get some bioasphalts and reduce the fire, the impact fire. Sorry, I forgot the name in English, but it's some of the principal cases that we work. And yeah, I think that's it. We are a startup. I hope we can continue with to finish our principal tools, AI tools.
Thank you, Sergio, for the great presentation. It is really exciting to see application of AI to biomanufacturing. So I'm wondering, in your perspective, what's the main challenge of applying the AI tools in this field?
Well, I think it's there is we have to develop it first some package of variabilities for all these kind of technologies, because as I said, I think the principal challenge is like every case is really unique. So the challenge using AI is to show to teach the AI how to identify that variabilities to make the real or the correct technical analysis on any case, I think.
Mm-hmm. Great. Do you find any difficulty, feeding, using enough or reliable data feeding to the AI, machine learning models?
Well, yeah, we are finding some difficulties now. I mean, it's a conceptual way now, but I think we can achieving. It's just we have to give a lot of database to the machine learning, and meanwhile we keep feeding the machine learning. I think we can make it easier with the time.
All right. I don't see any more questions in the chat. So I would like to thank you, Sergio, again for your presentation, and we can move to our last but not least speakers from Boston Bioprocess. Our speaker is Johannes. Also, do we have a second speaker?
Yes, Anupam is here. He is waiting. Oh,
welcome both.
You can yes, right?
Yeah.
Good.
So you can take it away.
Okay. Okay, I will share my slides and get started. My name is Johannes Kung. I'm the CSO of Boston Bioprocess. Let me see, does that? Okay, can you? I hope you can see the slide. Yeah, yes. So we are a small company, about 2 years old, located in central Illinois in Champaign. We are a service business for closing a gap, and you can kind of summarize that as enable and scale. We are about 14 employees at the time and constantly growing. And I kind of want to walk you through a little bit what we're doing, what is the gap I'm talking about, how can we scale, and then Anupam after that will talk a little bit about how we store our data and the whole how does that all kind of connects with the bioprocess side. So I kind of want to start with this slide and kind of point out that biomanufacturing is hard. There's no doubt about it. You have to first make a strain, and the strain is kind of your key IP that you have, and that decides a lot of things, but that is not where it ends. You kind of need to select a strain based on what kind of gives you the lowest cost, what works with the regulatory agencies, what is actually feasible to develop in the time you want, and what actually works at the high cell density. If you have something that you want to grow to a high cell density like yeast, E. coli, we mainly work with yeasts, bacteria, and filamentous fungi. Our clients mostly come from agriculture, food, and just industry applications. We have a tiny bit of pharma, but mainly we are in the non-pharma space. When you have your strain evaluated, then comes you kind of this is a strain I want to go forward with. Then you need to develop your process, and you need to really crank out as much product as you can per dollar that you put in. And you need to test, you need to revisit, you need to plan, you need to analyze, you need to establish really all the single unit operations. And when you have done that, you usually go to scale and say, okay, now let's see how this actually behaves in a production environment scale. And after that, you actually then are finally at that step where you can go and make product and make the money. That is a very, very long timeline, and there's a lot of pitfalls in there, and that's why a lot of companies start on the left, and not very, very many make it to the right. And this is a gap that we kind of identified, that I kind of want to highlight here on this slide. This is the 3 steps in between. There is a fantastic strain you made, and you want to get all the way to the other side, but there's a lot of things that might not be the expertise of the company, and really not the focus, because you just want to make the product. So you can take these 3 steps and develop them internally. You can buy your own fermenters for the bench scale fermentation. You can try all these things out, but there is the pain points that it is very expensive to buy this equipment that you might only need one time, or let's say 3 months, and then you're kind of ready for the next step, depending on how good you are. And that's kind of the second point. How much experience do you really have with the process development, with selection of the strain? You would have to hire people that you need to train, you need to commission equipment, and then that takes time, and then you are finally to the point where you can do really good development of your process. Then the pilot stage is very often disconnected from that. You wait for a while. When you go to a third-party developer, you pay a lot of money, but they don't know your process. They don't know your microbe. They are more familiar with maybe the scaling, maybe the equipment that they have, but they don't sometimes not really understand your process. They are not part of the development. So there's this disconnect. And then there's, of course, the transfer later on, where all of these things with analytic, with the strain understanding, needs to be transferred to the manufacturer if you're not doing that yourself. And that
sorry for the interruption. You are not in the presenting mode. Would you like to switch to the presenting mode?
Oh, sorry. Is that better?
Yes, thank you.
Okay, so we see ourselves as a business that makes scalable solution beyond assets, and that really means we are not just providing the assets and say, hey, you can come to us and tell us what to do, and we will run it, and then you might like it or not. We really put a heavy focus on, we will help you wherever we can to close the gaps that you don't know, that you have, where you don't need to know all of the little details about how to get to the market. So we don't just offer the assets, but heavy inputs on the development and on all the open questions that there might be. So we have the subject matter experts for the strain evaluation, process development, and the scale up. We have a full suite of equipment where we can choose together with the client what is the best choice for this process. And at any of those, at all of this time, there's no concern about IP or license. Whatever is developed for the client is owned by the client. So really, to get to the best process in the shortest amount of time, we are constantly reducing the cost for the process while we develop. We pride ourselves of a very fast, efficient, reliable, and thorough tech transfer to the large-scale production scale. But because we have the assets from the bench scale to the pilot scale, we can already make during development product that can be used for product development, application development, formulation, whatever else open questions there are that we don't cover. And that because we have the strain evaluation, the process development, and the scale up in one house, that usually leads to significantly reduced timelines versus other options where it's either the fermentation or the downstream development, or this scale versus the larger scale. Usually we offer the services in a pay per per batch mode. But if there's other financing issues, we can always talk about other options. In general, we promise that we will do the whatever we can to improve the yield, decrease the cost, and make sure that there's already product during development to do other testing. What that means, kind of to reiterate, we see ourselves really as something that looks at the process in the beginning like it should look at the end. So we already, when we do the strain evaluation, we look at what really counts at the industrial scale. What is the KPIs? Not because it looks good at the small scale, but what would it actually be at the larger scale? How would the cost actually look at the larger scale? Is this tighter doable at the larger scale? And it really helps that we don't just have the assets, but we have the people who know the product types and the organisms that are included. So we have a team of engineers and scientists that are really like interdependent. We have an expert for each step. When we talk about fermentation, we can do batch fermentation, fat batch fermentation, continuous fermentation, the typical aerobic fermentation to produce at a larger scale with the economical KPIs. And I think what I want to point out especially is that when we develop a process, or kind of how we solve some of the scaling issues, is that we already at the bench look at what actually works and what does not make sense. And with especially focus again on when you separate fermentation from downstream development, you usually have a really good upstream development, and then the downstream people need to figure out how do they solve these headaches that were created. So we really keep the downstream in mind when we develop the upstream, and that makes it much easier to use our expertise in the downstream purification afterwards, because we run into much less challenges as well. Whatever you put in, you need to take out afterwards, and that is something that is very often underestimated. We have experience with kind of enriching and purifying products that are hard to catch, that are maybe contaminated, or like not contaminated, like enriched with a lot of impurities that are hard to get rid of, because very often we don't have the problem, they're already gone when we come to this stage. And because we have all these steps in-house and we know all the steps at the same time, we have a very seamless internal tech transfer from the bench to the pilot. We are a very tight-knit team that always constantly talks to each other, so we make sure that we are up to speed on what needs to happen later. We have a platform for very quick churning out tech transfers into 4 CMOs, and we have already gotten a bunch of really good feedback from CMOs that say, hey, this is a really surprisingly good tech transfer. Everything is there and it's on time. And this is because we have good connections into the CMO space. We know what's out there. We know what's doable and what's not doable, which even means that when we develop things, if you already know as a kind of a product company what you want to do, where you want to go, then we can already start discussions, what's possible there and what's not possible there. Quickly, a slide with what we have. We have fermenters at bench scale. We have 16 1.5 liter fermenters. We have 3 15 liter fermenters, and then it starts with the stainless steel 20 liter, 75 liter, 500 and 1500 liter. And for the downstream, of course, we have a floor centrifuge for the bench scale. And then we have a Clara 20, Clara 200, and a nozzle type GR centrifuge. For the disc deck centrifugation, we can use decanter and ceramic membrane filtration just to separate the cells. If the product is in the cells, we can use high pressure homogenizer or bead mills or chemical lysis. And then to purify and clarify the whole soup, we can use dead end filter like plug flow or TFF with spiral wound or flat sheet. That really depends on the application, on the membrane availability, the scale. of course, and we have a few options there at different scales as well. So we don't always have to go to the 500 or 1500 liter right away to test this stack or spiral wound. And we are in the process of onboarding chromatography because we realize that there is more and more demand in the space for that. We can extract from solvents with a large pilot scale solvent room. And of course, we have drying capabilities, freeze drying, spray drying, oven drying, and tray drying. And throughout the whole process, we can offer a bunch of analytical methods. Like during fermentation, we have a CDEX and a YSI or HPLC for metabolites or the products. We can do protein enzyme assays, UV-Vis, SDS page, all of the typical analytical equipments or methods, and we have a really well-established relationship with Mario and Eurofins for all the things we don't have. So I only have a few pictures here added. I saw that Bruce is online from TJX. So at the bench scale, we have this really nice system, the 1.5 liters on the very left, and we have glass fermenters for 15 liter, 20 liter fermentation. Here is just a few illustration pictures. Here's the downstream with our stainless steel TFF skid from GIA for CMF or spiral wound, our Clararod 20 and a homogenizer, and a tiny picture of a bench spray dryer where we can already develop with very small samples and kind of see that is the product stable when spray dried. Does it even make sense to move forward, and what would be the conditions then we can adjust at the larger scale. And yeah, I mentioned the analytical capabilities. This is just a little bit illustration. We have a young, very motivated team that works tirelessly to make this happen. So we are very fast as well when it comes to onboarding compared to other options out there. I want to finish with one slide with a testimonial. We have more, but this is really one of the first we had that kind of is super happy with us and shows that it looks like we really help them make something work in a timeline that they needed. I would stop here and take the questions after the part from Anupam, who I will hand over to now.
Hi, I just want to spend the last few minutes to kind of discuss the other side of the solutions that we provide. So as Johannes mentioned, that we are not just an assets company, we see ourselves as a solutions company where we provide solutions obviously on the hardware side of things, of how do you develop your process and scale them up, but also on the software side of things. Because nowadays, data is very important in making decisions, and we are building an ecosystem within Boston Bioprocess to help our clients make more sense of their strain, they make more sense of their fermentation data so that they can excel in the product that they're making, but also in the scale up that they want to do. So very quickly, what our philosophy is that we want to provide our clients as much information as possible from us so that they can make the best decision. So the way we do it, that we are building a software ecosystem that we use to track all the data that passes through our unit processes. So for example, for every transfer is tracked in our system. We have dashboards for visualization of the parameters that you see for individual strains and the processes that goes on with it. That helps obviously in the data security and the information dissemination that we have, but also because we want to do more things through a digital framework, we want to reduce the human errors that naturally happens when we are doing things manually. We use these dashboards to also gather or generate insights so that you can actually, as a client, but also using our expertise from our engineers and the scientists, compare the performance of your strains for different runs. So as Johannes mentioned, we have many bench scale fermenters. So if you're running multiple fermenters using slightly different parameters, you can compare the performance of your strains at the fermentation as well as at downstream stages from one set of parameters or one strain versus another. And also that helps us develop analytics or analysis of how it is performing, how each parameter contributes to how the performance of your strain is, and not only just at the fermentation step. Because we are end-to-end bioprocess, we also want to include the downstream effects of your fermentation parameters into account so that you can actually perform an analysis in a full holistic manner. So what you get as a result is not only the expertise from our scientists, but and the wet lab and the process development, but also from our software tools and the statistical methodology that we use to kind of analyze these things. And so that was more on the operations side of what we are building. But the way that at the backbone of all of this that we are slowly building up internally is what I consider, quote unquote, a digital twin. So that digital twin is basically trying to gather the information to perform simulations both at the process development scale so that you can actually simulate how you can change different parameters and see different effects. And can you optimize your process development at the bench scale? You can actually then use that to evaluate what is the best set of parameters or the best conditions or the best set of downstream unit operations that you can use to get the most out of your product, as well as that will be used also in the process scale up. That how much can we actually use this information, because we have the kind of unique advantage of both having the bench scale, the medium scale, as well as the pilot scales in the same, in our same company and same data ecosystem. You can actually see how your parameters affect as you scale up, and that can be used as you kind of grow up to more CMO scales, so that you can get the best out of your strain for your product. And with that, thank you for listening to us. I am Anupam Chowdhury. I'm the head of data science at Boston Bioprocess. Johannes is the CSO. Please reach out to us if you have any questions regarding how we can actually help improve your process, develop your process, scale up your strain. We are happy to help and happy to chat. And with that, if we have any questions, we're happy to answer.
Thank you both. We do have one question in the chat for Johannes. What's the largest scale you have transferred the process from your pilot reactors, which is 500 to 1,000 liter to so far? So beyond your pilot scale reactor, what's the largest scale you have transferred so far? Did you see any performance degradation?
So the largest scale, that's easy, that's 10,000 liter. We have not we haven't really seen the data that how it performs. So let's say the client didn't really come back and say, hey, this works perfectly. We know it's working, that's what we know, but we haven't really compared and we haven't been able to see how it performs. But I mean, based on our development and how it went from bench to the pilot, I have no doubt that it will perform equally well. So let's say we have not had any complaints.
Great. Any other questions for our speakers? I have a question. I really appreciate your point on considering the downstream recovery upfront while developing your fermentation process. So could you tell us a little bit more how your team worked together on that towards that goal?
Yeah, it is really, so like I mentioned, we are a pretty tight-knit team. We are a pretty small team, and that makes a lot of things easier. So we are not siloed into a lot of different departments. We have a lot of people who have worked at all scales and upstream and downstream processes. I think we are in the process a little bit where it splits up a little bit, because it's obviously more efficient when you split this up. But I want to avoid this as much as possible, that people suddenly don't know anymore how this works. So everyone in our company has run TFF and fermentations equally, and they know exactly that when you have a certain pickier strain, let's say, that if you do not think about it ahead of time at the fermentation, that you will run into issues when you concentrate. It will, the flux will decay massively, and you can avoid that. You just need to talk about it from the get-go. You kind of need to know, okay, this is this kind of pickier strain. Let's think about what that means for the downstream. How can we, and the upstream, by media selection, by growing conditions, you but still making best use of the promoter. Make sure we get the like the titer, let's say, develop a compromise of what's the best titer with the least headache at the downstream, because it doesn't help you if you get more product in the upstream and you spend significantly more money, membrane area or whatsoever, in the downstream. That is a wash. You can think, or even worse, you can kind of maybe make a little compromise, get a little bit less in the upstream, but have a significantly smaller headache at the downstream. So that we have meetings where we talk about it. This is our new client. Let's sit together. What's the key points here where we need to talk about, that we need to keep in mind? What is doable at the larger scale? What can we not do? Where should we not go? There's the typical problem is clients come with us, have their strain, and they say, yeah, this strain was developed, makes 10 grams per liter. And first thing we say, what was the system it was developed? Because most likely it only makes 3 or 4, because the 10 grams per liter is under conditions that are not reasonable or feasible at a production scale. Something like, I don't know, 2-3 ppm air is just not something you want to do at a 100,000 liter scale. So that we discuss this, what is the goal, what is the minimum, and what do we need to think about before we even start the bench scale development or strain screening. What are we set ourselves our own filters where we say, we cannot go higher than this. We know our OTR at this scale, at the larger scale. Where's the problem? How would it look like at a 100,000 liter fermenter? So let's not develop something that leads to pain and disappointment, because obviously our number one goal is we want clients to kind of graduate, go somewhere and say, hey, go there. These are the people that really made my life easier, not wasted my time, and I have something that works.
Thank you. Yeah, we have next question for Anupam. Is your digital twin purely based on statistical model, or is it hybrid?
Hi, I responded back in the chat, but happy to discuss here as well. So in effect, it's a combination, right? It's a combination of mechanistic models when mechanistic principles hold, as well as data-driven and statistics-driven parts when we do not have enough information and we want to kind of let use of the data that we are generating internally and through mining of literature and other processes to guide how the simulation and the model will be built. So it's a hybrid one. We want to basically make use of whatever is the best technology available for the right situation.
Thank you. I don't see more questions. I think with that, we can conclude our session. And thank you all to our speakers and for the great talk. And thank you for all the participants for sticking with us for this 4+ hours. Really appreciate your time.
Thank you. Thank you, speakers and Ning, for moderating this session. It was an excellent session with lots of very important knowledge. I really hope entrepreneurs everywhere take note of this knowledge, because that hundreds of thousands of work hours, man hours, or people hours put into this to make this happen, to make this knowledge available to you all.